Oh, hello, 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 hello.
Hello, hello, hello. Hello, 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 hello. I request everyone to maintain.
Hello, 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 hello.
Check. Hello.
शक्ल भी आ गई है हटानी पड़ेगी घर वालों को भेज दी गई है ठीक है सर ठीक है सर वो हमारी इंट्रो में ही है मतलब वी आर इंट्रोड्यूसिंग कि लाइव स्ट्रीमिंग होगा साथ साथ
बस बच्चों को बीच में बोलते हैं कि मेंटेन करो साइंटिफिक कुछ कुछ बोलते हो अगर बोल सकते हो तो बायोइनोवेट के बारे में
पूछा था सर वो मना कर दिया गया था मतलब मैम ने बोला था कि ऑडियंस एंगेजमेंट की जरूरत नहीं ऑफिशियली शुरू करना है एक बार I request everyone to kindly take their seats as soon as possible and maintain the decorum in the Shanti Devi Mittal Auditorium.
प्लीज मेंटेन डेकोरम एंड व्हाइल द गेस्ट एंटर यू ऑल
Hello, 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 hello. Hello, 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 hello. Hello, hello.
हेलो हेलो
Hello, hello.
Hello, 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 hello. Hello, 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 hello. Hello, 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 hello. Hello, hello.
Hello, hello, hello.
Hello, hello, hello. The guests are arriving very soon, so you are requested to please maintain the decorum of the auditorium. Hello, 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 hello.
I urge the people, students who are sitting on the upper stairs, kindly maintain silence. It has become really very, very embarrassing at times in front of external guests. People, they don't stop talking. Please maintain silence and decorum.
ठीक है बट उनका वेलकम Ladies and gentlemen, I request all of you to kindly give a standing ovation with a thunderous round of applause to welcome our esteemed dignitaries and distinguished guests. They are present today as immense grace and prestigious event. As they enter, let's celebrate this moment that brings together some of the finest minds, leaders, researchers and visionaries all uniting under one roof to celebrate innovation, science, and collaboration. Thank you so much, everyone. We extend a warm and heartfelt welcome to all our respected dignitaries, speakers, and guests, and respected Pro Vice Chancellor, lovely Professional University, Dr. Lavi Raj Gupta, sir. We also welcome Head of School of Bioengineering and Biosciences, Dr. Neeta Raj Sharma, ma'am. We welcome all the heads, faculty, students, and participants of lovely Professional University. Your presence truly elevates this occasion and inspires every young researcher present here today.
BioInnovate 2025, organized by the School of Bioengineering and Biosciences, Lovely Professional University, is an international conference centered on the theme Bridging Science and Society for Sustainable Development Goals. This conference brings together researchers, academicians, and innovators from across the globe to share ideas, present breakthroughs, and explore how science can drive sustainable progress. I would also like to inform our dear audience and esteemed dignitaries that BioInnovate 2025 is running live on the official YouTube channel of School of Bioengineering and Biosciences. With that, let's begin this inspiring day dedicated to innovation, collaboration, and discovery. Ladies and gentlemen, in alignment with the vision of BioInnovate 2025 and our collective commitment to sustainability, we extend our warm welcome to the distinguished dignitaries with a green token of appreciation. We begin by requesting the respected Pro Vice Chancellor, Lovely Professional University, Dr. Lavi Raj Gupta, sir, to present the green token of welcome to the chief guest of the conference, Dr. Zameer Ahmed, sir, a Director, CSIR, IIIM Jammu.
Ladies and gentlemen, please give a huge round of applause as our esteemed guest is being presented with a gesture of gratitude and growth.
Now we may request Dr. Lavi Raj Gupta, sir, to present the next green token to Dr. Anil Kumar, Staff Scientist, National Institute of Immunology, New Delhi.
Ladies and gentlemen, please put your hands together as we extend this green welcome to our respected dignitary.
Thank you so much, sir. We now request Dr. Neeta Raj Sharma, ma'am, Head of School of Bioengineering and Biosciences, to kindly present the green token of welcome to Professor Shama Hiramat, co-founder and CEO, Lab Director, Trident Diagnostics and Healthcare Private Limited, Bangalore.
Dear audience, जोरदार तालियां हो जाए हमारे गेस्ट के लिए, ऐसे पल बार-बार नहीं आते.
Now we request Dr. Neeta Raj Sharma, ma'am, to present the next green token of appreciation to welcome Dr. Ajay Soni, President, ACBM.
आपकी तालियां ही इनके लिए सबसे बड़ा सम्मान है, ladies and gentlemen.
Now we request Dr. Himanshu Singh, Head of Department, to extend the green token of appreciation to welcome Dr. Girish Gaur, Associate Director, Rivara Labs, Mumbai.
Ladies and gentlemen, please give a huge round of applause as our esteemed guest is being presented with a gratitude of growth.
Now, I would like to request Dr. Himanshu Singh sir again to present a green token of appreciation to Dr. Rashmi Rana ma'am, Associate Professor, Department of Biotechnology and Research, Sir Gangaram Hospital, New Delhi.
जोरदार तालियां हो जाए हमारे गेस्ट के लिए.
Thank you so much, sir. Now, to honor the presence of respected Pro Vice Chancellor, Lovely Professional University, Dr. Laviraj Gupta, sir, I would like to request Dr. Neeta Raj Sharma, ma'am, to present sir with a green welcome.
Ladies and gentlemen, please give a huge round of applause as it will make this welcome more greener and warmer.
तालियां रुकनी नहीं चाहिए.
आपकी तालियां ही है जो इन पलों को और खास बना देती हैं.
Thank you so much, ma'am. Thank you so much, sir. With this, we conclude the Green Welcome ceremony, which reflects our shared belief that innovation and sustainability must always grow together.
Now, to mark the auspicious beginning of this conference, we proceed towards the lamp lighting ceremony, a symbol of knowledge, wisdom, and enlightenment. I would like to invite our respected Pro Vice Chancellor, Lovely Professional University, Dr. Laviraj Gupta sir on stage, along with our honorable dignitaries, Dr. Zabir Ahmed sir, Dr. Anil Kumar sir, Professor Shama Hiremat ma'am, Dr. Ajay Soni sir, Dr. Girish Gaur sir, Dr. Rashmi Rana ma'am, and HOS, Dr. Neeta Raj Sharma ma'am, and our respected HOD, Dr. Himanshu Singh sir, Dr. Ashish Vyas sir, Dr. Umesh Gautam sir, Dr. Joydeep Datta sir, Dr. Tejasvi Pandey ma'am, and our administrator, Dr. Munish Mehta sir. Ladies and gentlemen, let's welcome them with a huge round of applause.
लेडीज एंड जेंटलमैन, इस दीप की लौ हमें यह सिखाती है कि चाहे परिस्थितियां कैसी भी हों, एक छोटी सी रोशनी भी पूरे वातावरण को आलोकित कर सकती है.
दीप प्रज्वलन का यह पवित्र क्षण नए विचारों, नए संभावनाओं और नए संकल्पों की शुरुआत का प्रतीक है. Ladies and gentlemen, please give a huge round of applause for all our dignitaries and esteemed guests. A heartfelt thank you to all our dignitaries. Now I request our respected guests to kindly take their seats. However, I request Dr. Laviraj Gupta sir to kindly remain on stage for his welcome address.
Now, to officially commence this international conference, we have with us respected Pro Vice Chancellor, Lovely Professional University, Dr. Laviraj Gupta sir. Sir, we now request you to kindly deliver the welcome address and set the tone for today's conference. Ladies and gentlemen, please give a huge round of applause as sir shares his inspiring words with us.
I would like to welcome respected Pro Vice Chancellor, Lovely Professional University, Dr. Laviraj Gupta sir, who is a visionary academic leader known for his commitment to research, innovation and interdisciplinary excellence. Sir, we may now request you to kindly deliver the welcome address and set the tone for today's conference. Please welcome sir with a huge round of applause as he shares his inspiring words with us.
Check. Thank you, children. Thank you so very much. Good morning, everybody. Are you feeling lazy? I feel so, you're feeling lazy. Good morning, everybody. Louder. This is not the way biotechnology should work, feel like and work like. Once again, good morning, everybody. At an onset, on behalf of the entire family of lovely professional university, welcome the chief guest of the day, Dr. Javeed Ahmed, Director, CSIR, IIM Jammu. Thank you so much for gracing the occasion. It might take me a little longer time to name each and every keynote speaker, the session chairs, and the invited speakers. So we, as a LPU family, welcome them with a good round of applause. All the keynote speakers, the invited speakers, and the session chairs. We need to welcome the delegates, that's you, the budding researchers. So give a good round of applause to yourself. When the idea was put forward to me that we need to have an international conference under the aegis of Dr. Neeta Raj, she is leading the School of Biotechnology and Bioengineering. We discussed about that what is the biggest pain area which the humanity is facing. Because when we talk about biology, we talk about humans, we talk about all the living beings. And when we were actually doing the brainstorming that what should we capture? What should be the thematic credo of this international conference? We all unanimously boiled to just one word, and that is SDG. Because sustainable development goals, they're very meticulously, they're very prominently and preeminently designed to cover each and every pain area which the humanity or which the human beings or which even each and every living being in this world on this planet Earth is facing. I must congratulate the organizing committee, Dr. Neeta Raj and her team. Give a good round of applause that they planned this BioInnovate around sustainable development goals. But then, I have a point to make over here. When you are looking to the world spectrum, you are not looking to the Newton's fringes slit. You are looking to the world spectrum because you are actually focusing on sustainable development goals which are ordained by the United Nations. So your scope, your span is global. It's not local, it's not regional, it's not national. It gives us immense responsibility that in coming 2 days, we should at least propose 2 or 3 such brilliant and wonderful ideas, not just to the country's leadership, but to the United Nations. Then only these 2 days would be meaningful. Are you all with me on this? Because let the world understand that in 2 days what LPU can do. You know that you and your colleagues have done wonders out there. So why not to utilize these 2 days and the brilliance and the wisdom of the August gathering you have of scientists and industry professionals from around to create a dialogue I'm not wanting you to craft a policy. I'm wanting you to create or pave a new dimension or new direction in one of the sustainable development goals, be it hunger, be it, quality education, be it any of the 17 goals. Take a pledge, and Dr. Neetaraj, this is for you, that after coming 2 days, there'll be a signed document on my table which can be placed to our Honorable Chancellor. He's the member of Rajya Sabha as well. So he can take that dialogue further to the national forum, and that can actually get translated to United Nations. If that is the goal, then only we'll be able to meet up the expectations of 2 days and people present here. Do you want to listen to my journey of biotechnology and all this bioengineering? No mundi line. Say it, you want to listen to my journey? Because I'll give you the technical aspects that how biotechnology has taken shape. Because when I began with biotechnology, presumably I was the first PhD holder of bioinformatics, and I came from a background of mechanical engineering, and I did my master's in computer science. And somehow, somewhere, it was injected in me, not inculcated, it was injected in me that why don't you take up bioinformatics so that you can create a PKI. It's known as public key infrastructure, which you all do with your card transactions and or your mobile transactions right now, using the DNA structure which is absolutely unique to an individual. My guide was very strict, and he asked me to do 11 auditing of the courses, 6 of them at IIT Delhi, 2 of them at IIT Bombay, and I did a lot of audited courses on biology to understand biology fairly very well. And I still remember my days at IIT Kanpur and IIT Delhi. There were IBM mainframe computers, and to do global alignment of sequences with just 100,000 MERS. The IBM computer used to take 40 to 45 minutes to give me the similarity index. I'm talking about just pairwise alignment of 2 sequences having 100,000 MERS only. And when I came back to my PCs, those were the times of x86, you call them 386 or 486 or 586. People who are younger than me can understand that fairly very well. It used to take the same sequence alignment, it used to take 1 hour and 70 minutes, 1 hour and 60 odd minutes, or maybe around 2 hours, to just simply give me the similarity index of both these sequences. I'm talking about pairwise. When it comes to multiple sequence alignments, it used to take possibly an overnight to do so, be it on the IBM mainframes also. And this all, Cluster W, we didn't got Cluster W till then. We developed our own algorithms and programs in Fortran earlier. Fortran was slower, so we actually converted them into C and then C++. And today, 100,000 MERS, you talk about 1 million or 1 billion MERS. Pairwise sequence alignment, less than microsecond. Now that's the power of technology. You are born in an era where you do not have to actually write, those times there were no mails, there were no nothing. You used to get the sequences in FASTA format on papers, ATGC written on papers, you will have to actually punch them on your punch cards. You will not even be able to think what is going to happen. If one of the punch card goes wrong, the entire stack of 3,000 punch cards is wrong. You are born in an era that your mobile phone can do complex proteomics calculations at the mobile station only without even touching to the edge or to the cloud anywhere else. Very recently, very recently, there's a development which is going to happen, and that is actually for you as researchers in biotechnology, and that is known as Willow chip. Google has come up with a Willow chip. Now that's a quantum chip. If presently you are defining or designing a drug, it can design the same drug 13,000 times faster. Meaning thereby that if in COVID it took maybe 6 months to 8 months to design the drug, now it will be done overnight. This is what is called an opportunity for you. This is something which should not let you sleep. This is something which the world is expecting from all of you out there. You are the ones who are going to define the humanity in a different way, be it in terms of biotechnology, bioengineering, or clinical microbiology, or microbiology, or molecular biology. You are the ones who are going to define the entire drug discovery and drug scheduling systems. And now the technology is with you. You have heard about LLMs? Say yes or no. Large language models? Chat GPT to suna hoga na? Okay. Now these large language models are all language models. They can answer you something on a prompt which is written on a language for the answer to be in a language only. They are not math intensive. Things like cloud or DeepSeeks, they are somehow, somewhere mathematically trained. Somehow and somewhere, not fully. Why can't you people think of large bio models in place of large language models, wherein that particular model is too specific and too particular about all what happens in biotechnology or bioinformatics or bioengineering? I have given you food for thought. Now it's all up to you that how do you take it up forward. But believe me, utilize the technology, hibiscus the technology, and the world would be at your feet. I want the best of entrepreneurs from this group sitting and taking leads and threads from the dialogue which is going to happen in coming 2 days. I wish this conference to be a grand success. I wish you all the very best. Good luck and Godspeed. Thank you.
Thank you so much, sir, for your inspiring words and for always motivating young minds to explore and innovate. एक बार जोरदार तालियां हो जाएं सर के लिए.
Ladies and gentlemen, we are truly honored with the presence of our chief guest of the day, the director, CSIR, Indian Institute of Integrative Medicine, Jammu, Dr. Zabir Ahmed sir, a renowned scientist with more than 25 years of expertise in metabolic disorders, rheumatology, and diabetology. Under his leadership, CSIR IIIM has made remarkable contributions to drug discovery, technology incubation, and translational research. May we now request Dr. Zabir Ahmed sir to first kindly address the gathering, and then we will move on to his scientific presentation. Ladies and gentlemen, please give a thunderous round of applause as we welcome sir on stage.
I started my journey early in the morning at 4. After traveling for more than 4 hours, I was feeling tired. But when I entered this vibrant campus of lovely professional university, I got energized, and the kind of academic and research atmosphere I have observed here, this is the core feeling which I am sharing with you, that it was a great opportunity for a person like me to visit this great campus. On this occasion, I take this pride to salute Dr. Laviraj Gupta, Honorable Pro Vice Chancellor of Lovely Professional University, his vibrant team, Dr. Neeta Gupta, Organizing Committee, staff members, students, and the delegates of this international conference, BioInnovate 2025, Bridging Science with Society for Sustainable Development Goals. From my side, from my institution, I am taking this opportunity to extend greetings, namaskar, and adab to you all. For a person like me, selecting as chief guest of this important event, for me, I think I am not fit for this honor. Anyway, for this recognition, for this honor, I am really thankful to the Pro Vice Chancellors, Organizing Committee, and other stakeholders of this important event. I have seen the theme of this international conference. You know, we have 17 SDGs. In a short while, I will be sharing my experience about only a single SDG from institution side, what we are doing. But for lovely professional university, I don't hesitate to say that this is the institution which is responsible for all 17 SDGs. Again, from my side, congratulations and greeting, and thanks to the stakeholders, the management, and the other staff members of this great university of this great nation. The theme of this event is meticulously crafted. Science and society. Are we thinking that there is a gap between science and society? For students, for other delegates and the learned speakers, this information is not meant for them. This is meant for students only. Dear students, there was a time when we used to say science is a subject, and we need to just adopt medical or non-medical, and preferably we just confine our feeling about these two streams only. Dear students, now it is the time that science has become way of life. You point out any activity, you point out any subject, any field in front of you, within the society, within the region, at national level, everywhere you will see the impact of science. And again, The question is that whether our stakeholders, we, the responsible people in science component, are we are in a position to bridge the gap which is having between science and society? When I say science has become a way of life, I also confirm that the kind of policies, the kind of projects, and the kind of programs institutions like scientific institution, academic institution, and the university like LPU, all these are responsible for bridging this gap, and up to some extent they are successful. When I say we are in a position to bridge the gap between science and society, I mean to say we are doing science. Like I am coming from an institution which is involved in drug discovery from natural resources. And where from these natural resources are coming? These are coming from fields. And who are involved in cultivations of the plants and natural resources? We people, our farmers. When our startups, the researchers, students, faculty members, scientists are involved in such kind of activities, our claim is justified that we are moving towards bridging this gap between science and society, and which is the critical component as far as implementation and successful achievements of SDGs are concerned. At the same time, I wish to share with students and other delegates that when we have all the stakeholders on board, we have researchers, we have industry people, academicians, scientists. At the same time, certain challenges are also there. And such kind of events, important events, where deliberations, viewpoints from different keynote speakers, other participants are held. So such events provide us the opportunity to come up with a policy document, to propose some improvisation in the policy document as far as the development goals are concerned, as far as overall development of nation is concerned. You know that we are into March of Viksit Bharat 2047, and our focus is to have Make in India in every sphere, whether we use food items, pharma, medicines, machinery, our focus is to produce indigenously. But when we talk about drug discovery, you will be surprised to note that we are the number one importers of APIs from China. Despite the fact that our country, this great nation, is having 6th rank in biodiversity scale. And if we talk about this region, like Himalayan region, we have biodiversity hotspot. This region is known as hotspot of biodiversity. It means that this region harbors more than 3,000 medicinal plant species. These natural products are responsible for producing these APIs, but at the same time, when I say challenges are there before the stakeholders, it means that when resources are available, when brain is available, when the knowledge is not injected, it is being inculcated, but at the same time, we are facing such problems. And this is the ultimate responsibility of all the stakeholders to look into, to bridge this gap in the innovation valley of death. We have researchers, good number of students are there, good number of researchers are there, good number of scientists are there. If I simply talk about CSIR, we have more than 3,500 scientists across India. And if you talk about the research scholars and other students involved in research, they are in lakhs. But we need to just have a clear-cut focus how we can bridge this gap. Ultimately, such efforts will definitely help us to bridge the gap between science and society, and if we really want to contribute towards SDGs. When I mentioned about APIs, many students, many researchers will definitely surprised to know that we were having these facilities in India. Pharma people used to manufacture such APIs. But when when these APIs were being supplied by China at very low cost, so such types of critical factors are there and we need to just look into that. So I mean to say, when we talk about implementation of certain guidelines, certain policies to contribute towards SDGs, to contribute towards Viksit Bharat 2047, such factors are very important. And what I hope the due deliberations during next 2 days will definitely contribute in a big way as far as bridging the gap between science and society is concerned. And one word, that is BioInnovate 2025. Coming from background of drug discovery, I again share this idea with all the scholars and students. When we are having these resources, when we are having the institutions of excellence, another very important component is that, like my institution, other institutions, we need to provide incubation support to all these innovative ideas as far as development of incubation of these ideas is concerned, because all researchers, all ideas cannot have that kind of facilities which you can avail from CSIR institution or the institution like LPU. So this is another important component we need to just look into. So on this occasion, from my side, from CSIR IIIM side, I take this opportunity to congratulate the organizing committee, the stakeholders, the delegates of this 2 days international conference. Definitely it will be a robust platform to deliberate, to contribute by innovative ideas, and to come up with a white document like Professor Gupta sir has mentioned, that we need to just submit one document to him so that the kind of research policies as far as our objectives in this international event is concerned. So definitely by way of such kind of deliberation, definitely we will be able to meet the basic mandate and objective of this August gathering, August meeting. So again, my sincere thanks to you. With this, the theme of the conference is BioInnovate 2025, Bridging Science with the Society. So I will just give a quick presentation where I would like to share with you the kind of research activities we are doing. And definitely from this brief presentation, you will come to know about our role as far as our contribution towards one of the SDGs is concerned. So may I have this presentation, please?
Dr. Ahmed will be delivering his presentation on the topic, Phytopharmaceutical Drug Discovery, an approach to globalize Indian traditional medicines, sharing his extensive research experience and deep insights into the advancements of biotechnology for sustainable health and development. Let's hear a huge round of applause for Sir as we look forward to his enlightening presentation.
So again, from this presentation, I would like to share a few ideas, few research component of our institution, and at the same time, I look forward for an interaction, a great learning for me. So again, Thanks to the organizing committee for giving me this opportunity to part of this very important event. So before I share few slides, I would request if any of my slide will go out of the subject, the theme, I beg my apology in advance and you will bear with me. Phytopharmaceutical drug discovery, an approach to globalize our Indian traditional medicine. When I used this word that we are having rich biodiversity, dear students, this great nation is having a rich legacy of traditional medicines. We have Indian system of medicines which is being practiced for centuries together. And when we are looking towards SDGs, we are looking towards health, wellness, and when we are looking towards the global competition. So definitely our policymakers, our government was very serious to enact this Phytopharmaceutical Drug Discovery Act in 2015. Before 2015, whatever Ayush drugs we used to work on, these drugs were from Ayurveda, Unani, Siddha, homeopathy. But with the enactment of this Act, Phytopharmaceutical Drug Discovery Act, now you will see the typical guidelines for drug discovery which are typical and similar to the allopathic system of drugs discovery. Before I go to the actual type of work we are performing there, I also take this opportunity to share a brief introduction about my organization and the CSIR. At Pan India level in CSIR we have 37 institutions. This is Council of Scientific and Industry Research under Union Ministry of Science and Technology. Honorable Prime Minister is ex officio President of this council. Honorable Union Minister for Science and Technology is ex officio Vice President of CSIR. And we have Director General who is Secretary to Government of India, Department of Scientific and Industry Research. When I mentioned about the presence, Pan India presence of CSIR, we have 37 labs and all these labs have different domain areas. Like we are talking about 17 SDGs. In a similar fashion, in a similar way, the classification of domain areas of these CSIR institution has been decided. We have different cluster, like biological cluster, about 10 labs are there which are working in drug discovery and other biological aspects. We have chemical science cluster, engineering sciences cluster, information science and physical sciences. All 37 labs have been divided as far as their domain areas and expertise of work is concerned. Coming to CSIR IIIM Jammu, from which I am representing this event. This institution was established in 1941, much before independence, as Drug Research Laboratory in Jammu. Till 1957 it was DRL. In 1957 it was renamed as Regional Research Laboratory. So at that point of time the main focus of this institution was to explore the natural resources from region from Jammu and Kashmir, from Ladakh. So a lot of work has been done, and You would be happy to know that the reserpine which we are using today, that drug has come from this laboratory only. Colonel R.N. Chopra, who was founder of this institution, he identified this molecule from natural resources, and this molecule later on developed as drug. In 2007, after broadening the mandate and mission of this institution, it was renamed as Indian Institute of Integrative Medicine. For the information of students, for the information of these scholars, I must say Indian Institute of Integrative Medicine. A while ago I mentioned that we have rich heritage of Ayush system, we have Indian system of medicines, and the core mandate of this institution is to integrate the traditional medicines with the modern medicines and modern drug discovery. So that's why this institution was renamed as Indian Institute of Integrative Medicine. We are talking about BioInnovate, we are talking about science. So I think in some or the other way this institution, CSI, Indian Institute of Integrative Medicine, is doing little bit work for bioinnovation. We have two campuses. Beside, if we talk about the natural resources, we have different geoclimatic zones in Jammu and Kashmir, and accordingly we have different experimental farms. In Jammu we have one big farm which is at Chatta. Then in Anantnag, the Varinag farm is there. In Bonra, Pulwama, 150-acre farm is there where huge lavender cultivation is being done. Then we have another farm in Yarika. And recently we acquired one land in Ladakh to start the cultivation of medicinal and aromatic plant. Hereinafter, whatever I will say, that will related to science and society. For society, for outreach, what kind of activities, what kind of programs this institution is having. And in science aspect, particularly in drug discovery, the kind of work our researchers, our scientists are doing, that will be shared. So if we talk about the specific drug discovery program, we are working in the area of cancer, infectious disease, inflammatory disorders, metabolic disorder, and neurodegenerative disorders. And we have different sectors: phytopharmaceutical, nutraceutical. On nutraceutical we are working, and beside this we are also working for medicinal chemistry, synthetic molecule preparation is being done also. So this is the outline of the research profile and the research activities we are doing at IIIM. As I mentioned, the Indian traditional system of medicine, traditional medicine system is very old, and it has a great value also. We in India are having, nature has bestowed this nation with more than 7,000 medicinal plant species. So this is evident, and this is sure that whatever molecules we are synthesizing today, whatever molecules, APIs we are manufacturing today, all these molecules are available in nature. And when we are able to explore all such molecules for different kind of disease targets, definitely we will be able to contribute significantly towards development of science, particularly the bioinnovation. As far as economy and the global figures is concerned, from India, export of medicinal plant annually, if we can consider, this is more than $110 billion. In India, you will surprised to note that more than 400 research institutions are involved in traditional medicine systems. When we are having rich brain, we are having a number of institutions, and we are having the rich resources where the medicines, raw materials, sourcing of raw materials available. So the requirement is that we need to just globalize our Indian knowledge. And that is the requirement of time. If we claim that we are really working towards SDGs, particularly for health and wellness, the stakeholders, the students, who are the real stakeholders for Viksit Bharat 2047, we need to focus our research in this area also. So at Indian Institute of Integrative Medicine, we have multifaceted approach for preclinical drug discovery. My dear students, for any drug which we take from market, we see drug is available in the market, the drug molecule, the medicine has to undergo series of activities in the preclinical phase and in the clinical phase. So when we talk about the preclinical drug discovery from natural sources, the composite model of preclinical drug discovery is available at CSIR IIIM. Bioprospection from nature sources, authentication of nature sources, extraction, standardization of extracts, isolation of molecules, upscaling, all kind of facilities are available. And this would be followed by the in vitro, in vivo assay systems for different disease targets. Like I mentioned, we are working in different disease areas. The assay model, the experimental models are available. So it means this institution is having a composite model for preclinical drug discovery. In addition to this, we are having collaboration with pharma industry, various institutions, and we have certain societal mission programs. When under these programs, we are working for society. So if we activity portfolio of this institution consider in international conference theme, two words are there, science and society, and in this slide you will also see two words. When we are talking about science, we want to become global competitor. In US market, you can see the phytopharmaceuticals are not there, botanicals are there. The US FDA guidelines are there to develop the drugs from botanicals. And parallelly, we in India are having this phytopharmaceutical program, and at IIIM, this institution is nodal in phytopharmaceutical drug discovery. AMR is another problem. So this mission is being implemented and we are partner in that. Another important research work, when we are talking about BioInnovate, when we are talking about research, and when we are having such resources, you might have heard about cannabis. Cannabis is a prohibited plant, and we have for the first time started research on cannabis, and a couple of leads we have identified which are moving towards clinical development. And with the society, for the society, our main focus is to empower the farmers, empower the society people, society members. We have aroma mission, we have floriculture missions, we have different kind of services which are providing to the society, to the startups, to the students. So through this component, we are helping our society to contribute, to involve themselves in bio-innovation for research, and particularly when we talk about, when I say that we are just committed to empower the farmers, through these mission programs, our farmers are empowered and their income is significantly enhanced through missions, these societal missions. We have collaboration with the industries. Many industries are working together with us. We have CGMP facility. Any drug which has to be taken forward for clinical development, so manufacturing of such drugs are done in CGMP conditions. So this institution is having CGMP facility also. This I have covered. We have very rich resources of plants and other nature resources. Biodiversity is available, world-class infrastructure is available. We have incubators from which we are providing support to researchers, to students and the startups. These are the main facilities for preclinical drug discovery we are having. For students, any drug molecule, when it will be moved towards clinical development, regulatory toxicological studies are done, and such kind of facilities, GLP standard animal house is available. Fermentation facility, BSL-3 facility, other we have another important aspect like natural product repository. So when we are talking about APIs, when we are talking about reference materials, if such kind of facilities are available, definitely we can move towards the import substitution. If we can really upgrade, we can really start some processing and we can develop some process for synthesis of these APIs, definitely we will be able to contribute towards the import substitution, and definitely it will add to SDGs contribution and add SDGs roles of the institutions. I mentioned about Indian system of medicines. We have Ayurveda, Yoga, Unani, Siddha. Recently, Sovaripa, the Tibetan system of medicine, that is added in Ayush. Homeopathy is there. And beside this, I mentioned about allopathic. In comparison to, similar to allopathic, we have this phytopharmaceutical drug discovery in India. So the key question is, we are working on natural products, we have the herbal resources, we have the research programs. So can we position these Indian traditional medicines into global market? What I observed, and as far as my knowledge is concerned, phytopharmaceutical drug discovery program is a strong link to modernize the traditional Indian medicines. So this I mentioned, like all kind of preclinical drug discovery facilities and the resources are available in the institution. Now I will talk little about the phytopharmaceutical drug discovery. What is phytopharmaceutical drug discovery? For years together, we used to have this Ayush system of medicines. The big challenge with the Ayush system was the quality control, the standardization, the authenticity. Everybody accepts this fact. In order to standardize these natural resources, herbal resources, Government of India in 2015 has enacted this Phytopharmaceutical Drug Discovery Act. And this is typical to the allopathic system of drug discovery. All the key components which are involved, all the key activities which are involved in preclinical drug discovery of allopathic medicines, these are included in the guidelines of phytopharmaceutical drug discovery. So outline of this guideline is that we need to have a standardized fraction or extract from plant source which is having at least 4 bioactive markers or the standardized markers, and it should be used orally or locally. Parental route for phytopharmaceutical drug discovery is not allowed. So this I already covered, like we are contributing very huge amount as far as the export of these raw materials of plants and herbal resources is concerned, and at this, if global burden we consider, it is more than $100 billion. And global requirement is, if you can see, but despite the fact that we are having a rich resource of these resources, these herbal resources, phytopharmaceutical, towards global competition, at the moment we are contributing only 1 to 2%. So regulations are available. If we compare the regulations of phytopharmaceutical with the US FDA guidelines for botanicals, These are comparable and we can compete in that. And as far as the global positioning of these drugs is concerned, definitely if we can do the phytopharmaceutical drug discovery research as per the phytopharmaceutical guidelines, we can meet the global standard. So this is just the outline for the information of the students and the learners. This is just the outline of the activities we need to do for phytopharmaceutical preclinical drug discovery, standardization of the fraction and extract on the basis of the markers, preclinical in vitro in vivo efficacy, regulatory toxicological studies in different animals, then PAPIs, that manufacturing is done in CGMP, stability studies, preparation of dossier and filing of IND. So this is the outline of the activities which we are required to do for phytopharmaceutical preclinical drug discovery. So this is the mission statement for phytopharmaceutical drug discovery as far as Phytopharmaceutical Act is concerned. As I mentioned, raw material is available, standardization is not there. Issue of quality is there. So in order to meet these deficiencies and these critical points, the Phytopharmaceutical Drug Discovery Act has been enacted. And at the same time, another important component in this aspect is involved, that is the empowering of farmers. When we are using raw material from farmer fields for phytopharmaceutical drug discovery, at the same time the beneficial effect, the benefit also goes to the farmers who are doing captive cultivations, who are doing cultivation of medicinal plants. So at CSIR level, we have this phytopharmaceutical drug discovery program. So far, we completed 2 phases, and if we typically talk about CSIR IIIM, these are the phytopharmaceutical leads for the first time in India we have developed, and these leads are at the advanced stage of preclinical and clinical development. You might have heard about saffron, crocus. This is natural. So from our lab, this is for the first time that we have developed a standardized fraction for the treatment of Alzheimer. IND has been filed for this phytopharmaceutical lead. Besides this, other leads are there for arthritis, osteoarthritis, and cancer pain. In addition to this, from natural sources, we have identified certain NCs, and these NCs, one is for pancreatic cancer, other is for triple negative breast cancer. So these are also at advanced stage of preclinical development. Like I mentioned, we are working with natural sources. We are in phytopharmaceutical, we are in nutraceuticals. Certain nutraceutical leads we have identified, we have developed, and technologies for these nutraceuticals have been transferred to the industry. So this is the outline of the activity we have so far performed in crocus for Alzheimer. So we are moving towards phase 1 clinical trial. We have filed the IND on the basis of the crocin content in this fraction. We identified it, and the important property of this lead is it is very potential in clearing the amyloid beta plaques from the brain. Besides this, it is also controlling the neuroinflammation. We have patent for this, and we are moving towards the clinical trials for this lead. Other lead is from disoxylum. This is for arthritis. Again, phase 1, we are planning to start with this lead. From Virginius Lata, we have program with DBT, and again IND enabling studies have been completed, and we are moving towards IND filing. You might have heard about pancreatic cancer. This is very critical and serious disease. As on today, we are only having injectable medicines for treating pancreatic cancer. This is for the first time that from CSIR IIIM we have identified pure molecules, CDK9 inhibitor. So we are into phase 1 and phase 2 clinical trial with these molecules. I mentioned that the drugs available are injectable. The potency of this drug is there, this molecule is there, and if the phase 1, phase 2 clinical trials will be successful, this would be first molecule from India which will be used orally. So this is very important lead from CSIR IIIM Jammu as far as pancreatic cancer treatment is concerned. For osteoarthritis, another, like I mentioned in previous slides, was from Boswellia serrata, this fraction has been prepared and we are moving towards the IND filing for this. So I mentioned about cannabis research. For the first time we started research in cannabis after getting approval from Jammu and Kashmir government, and we have identified the fractions. For the information of the students and the learners, cannabis had been used for centuries. When the British rulers left our country, so they had put this plant in prohibited class. When they were ruling our country, the plant was being used openly. They felt that they were having that kind of knowledge. They felt that this nation, this country will have a great impact as far as the research and the development from cannabis is concerned. They put this plant into prohibited category. And as on today, in India, this plant is in a prohibited category, but in US, Canada, and other countries, it is legalized. They are using medicine from these plants, this cannabis, but India, we are having this kind of restriction. Why I am sharing this? Like, if we are having this kind of deliberations during this very important event, so some document can be prepared from your side that we can put before government for a policy document for policy making on cannabis. From our institution, we have already submitted one document to Government of India that in India we should have a policy on research on cannabis, so that for research purpose, for medicine purpose, we can use this plant. And the wonder of this plant is that in single plant, we will find molecules for 5 diseases. So you can imagine the importance, the significance of the composition, the constituents of this cannabis. So we completed the preclinical research and we got the patent, and we are moving towards the clinical trials. And for this, again, we approached the Jammu and Kashmir government and central government to grant us the permission to start the GMP manufacturing and the clinical trials. In continuation to the phase 1 and phase 2 of the phytopharmaceutical mission, we have now phase 3 phytopharmaceutical mission. On one side, we are doing drug discovery. On other side, we are helping our farmers to grow the medicinal plants in captive conditions. So here we are helping, empowering these farmers to make best use of their land, to grow medicinal plants, so that the raw material chain can be established. And this is very important program we are running from CSIR side. So this is the DTL facility we are having, so I need not to just mention about this. This is the common facility. I understand the students and other audience got bored. I will take only 2 minutes and I will leave the stage. In the outreach component, for the benefit of the society, from CSIR side, we are implementing CSIR Aroma mission, where more than 2 labs are involved. Different aromatic crops are being cultivated by our farmers. And for the cultivations of these aromatic crops, right from providing the quality planting material to agrotechnology, distillation of essential oils, processing of these oils, quality control, product development, market linkage, we are providing. So in this aspect, this is the typical example where we are connecting our research with the society. On one side, we are providing support to our farmers for increasing their income. On the other side, we are doing science to improve the quality of the aromatic crops, quality of the aroma chemicals present in these crops, and at the same time we are identifying the medicinal components available in these aromatic crops. This is the real example of connect between research and the society. In addition to this, another one thing I would like to share with you, in Aroma Mission, you might have heard about this purple revolution in lavender cultivation, and this was implemented in Jammu and Kashmir region. So for students, for scholars and startups, just a single example I would like to just cite. We started from one flower when we introduced lavender cultivation there. We have more than 3,000 farmers who are involved in lavender cultivations. There was a time one farmer came to us and he showed the reluctant to start the cultivation of lavender. We motivated him, we've provided all kind of support, and it was only during last month 2 farmers returned back to us and they become industrialists. They are saying we are in product development, just sign some MOU with us so that we can jointly produce some marketable products from the aroma crops. So this is an idea, this is an example where we can see if we are having certain ideas, we can use our lands, we can use our knowledge, we can get the support of the incubators, the institutions like LPU, and we can develop. On one side, this will be a support to the economy of the farmers. On other side, we will be actively contributing towards nation building and Viksit Bharat. Another mission we are implementing, that is floriculture mission. To cite the example of floriculture mission, what we are doing in Jammu and Kashmir, annually we are importing flowers worth more than 20 CRs. When we started this mission in Jammu and Kashmir, according to the estimate provided by Agriculture Department, our farmers are contributing the flowers worth 3 crores annually. Their target is 20 crores. So with the help of our institution, they are growing these floriculture crops and they are doing business. This is not only the end. What they are doing, they are selling these flowers. Then since Jammu is known as city of temples, many other places are there where these flowers are offered. Our research team is getting back the waste flowers. They are involving these farmers only to produce certain perfumery products or agarbatti, then the flavoring agent they are making out of these used flowers. So it means end-to-end utility of these produce, these crops are being done, and ultimately in one or the other way, we are helping these farmers to increase their income. I mentioned about saffron. We extended saffron cultivation in Leh and we got the success. So this is little important, like when we mentioned there should be a connect between students, scholars, institutions, scientists and academicians. Such kind of platforms provide support to the incubators and the startups. So we have 3 incubators, Technology Business Incubator in Jammu, Bioness Incubator in Jammu, and Atal Incubation in Kashmir Center, where we are providing support to students, farmers, industry people. We are imparting skill development courses to the scholars and the other students. So it means, as far as concerned, as far as connection of our society with the institution, with the research institution and academic institution is concerned, such kind of incubators provide a robust platform for students, for researchers, for academicians, scientists, industry people and policy makers to come together, to deliberate and to contribute towards the knowledge development, incubation and the policy development. I think I have bored you well, so you will excuse me for that.
Let's hear a big round of applause for sir.
Thank you, thank you. The kind of hospitality, the kind of reception, the kind of honor I received from this institution. This is really, really great moment for me, and I will remember this moment. I will definitely cherish this moment. With all these words, I also take this opportunity to welcome you all to my institution. Whenever you will find any time, any opportunity to visit Jammu, just visit CSIR IIIM Jammu. Definitely you will practically and you will personally see the kind of facilities about which I have talked during my presentation. Dr. Neeta Raj mentioned about the kind of collaboration, the kind of joint program. Like I mentioned, we have many partnership programs with industry, with institution, with government institution, with private institution. Really, this is the great opportunity for me to invite lovely professional university to join hands with CSIR IIIM Jammu so that actually we will contribute towards nation building, the requirement of the society, the requirement of the science and requirement of Viksit Bharat 2047. आपका बहुत बहुत शुक्रिया, धन्यवाद, जय हिंद.
Thank you so much, sir, for your insightful and thought-provoking presentation. Your ideas and experience have truly added a meaningful dimension to this conference. Let's hear a huge round of applause for sir. We now invite the head of school of bioengineering and biosciences, Dr. Neeta Raj Sharma ma'am, to deliver her vote of thanks and express gratitude to all the dignitaries, guests and participants who have graced this occasion. Let's welcome ma'am with a huge round of applause.
Check. Great morning, one and all. Good morning. So it's indeed my pleasure, privilege, and honor to propose the vote of thanks on behalf of the entire organizing committee for today's inaugural session of the international conference entitled BioInnovate 2025, Bridging Science and Society for Sustainable Development Goals. The conference has been organized by the Department of Biotechnology, School of Bioengineering, Biosciences, lovely professional university, in collaboration with the knowledge partner, the Association of Clinical Biochemists and Microbiologists, ACBM. So first and foremost, I would like to express my deepest gratitude to our most revered chief guest, an academician, distinguished academician, eminent researcher, Professor Dr. Zabir Ahmed, Director CSIR, Indian Institute of Integrative Medicine, IIIM Jammu, for gracing his presence and sharing the valuable insights that have set the tone for this conference. Sir, we are truly inspired by your words and very, very motivating, encouraging examples you have given to us, to our students, and we are looking forward to your support and hand-holding from IIIM Jammu for all of us in our future endeavors. We are deeply thankful to our Honorable Chancellor, Dr. Ashok Kumar Mittal, Member of Parliament, Government of India, and our worthy Pro-Chancellor, Dr. Rashmi Mittal ma'am, to their constant guidance, motivation, and encouragement, Their vision and commitment towards the global engagement have been the driving force behind such international academic initiatives. Our heartfelt gratitude also goes to our Pro Vice Chancellor, Professor Laviraj Gupta, for his steadfast support and encouragement at every step of this great event. Students, you were listening when Dr. Laviraj Gupta was telling us about the presentation of one paper after the completion of this conference. And all these students of bioengineering and biosciences are already aware that they have been given one task, that they have to extract the highlighted points from all the keynote speeches, and they have to create certain life projects for them so that those could be workable. And as per the talk of Professor Zabir, he already gave us, or you can say that extended the help and support to all of you. If you have any project in mind, and if you are involved in the development of any kind of product, that can be further materialized with their help. So this is the responsibility now, that just mere sitting in the hall and just passing the time is not the objective of this conference, students. So you need to listen each and every talk very carefully and extract your points. You can write the review article, or you can design your own project that you can use for the entrepreneurship or the startups in the area of the medicinal plants. Are you all ready for that? Fair wahi, yes. How many of you are ready for this, to take this challenge? Raise your hand. Aise nahi. Raise your hands. So this side, students are sleeping. Students, raise your hands, I tell you. Very good. Thank you very much for your interest and enthusiasm. A big round of clap for you. Yes. A special note of gratitude goes to the knowledge partner, the Association of Clinical Biochemists and Microbiologists, whose collaboration and intellectual support have been instrumental in laying the foundation of this conference. And we extend a special thanks to the President, ACBM, Dr. Ajay Soni, President of ACBM, for his collaboration, partnership, and unwavering support since beginning, when we started the planning the title and the logistics, each and every the phase of planning of this conference and the execution, he was there. Thank you very much, Dr. Ajay. It is indeed our pleasure and privilege to have with us an eminent personalities from the field of life sciences, from industries, and from academia, from India and abroad. As our keynote and invited speaker, I would like to acknowledge their names over here. So we extend our sincere thanks to Professor Nasreen M. Hashim, Faculty of Agriculture, Alexandria University, Egypt; Dr. Indu Sharma, Research Scientist, University of Southern California, US; Dr. Robin Joshi, Staff Scientist, Institute for Translational Medicine and Therapeutics, University of Pennsylvania, US; Professor Shama, Co-founder and CEO, Trident Diagnostics, Bangalore; and Dr. Girish God, Associate Director, Rivara Lab, Mumbai; Dr. Siva Adarsh, Senior Manager, Neogen Labs Private Limited, Bangalore; Dr. Anil Kumar, Staff Scientist, National Institute of Immunology, New Delhi; Professor Shashank Deep from IIT Delhi; Dr. Pallavi Singhal, Scientific Officer, BARC, Mumbai; Professor Imtiaz Hassan, Jamia Millia Islamia, New Delhi; Professor Gulshan Kumar, Dean, SDS, Uttarakhand University; Dr. Ramesh Chandra Kasna, Principal Scientist, Microbiology, CFET, Ludhiana; Dr. Alpa, Consultant Biochemist, Hinduja Hospital and MRC, Mumbai; Dr. Varun Sharma, Head R&D Division and Bioinformatics, NMC Genetics, India Private Limited, Gurgaon; Dr. Sandeep Singh, Senior Scientist, CSIR, IITR, Lucknow; Dr. Anita Kamra Verma, Professor, Delhi University, Professor Manoj Banwal, Thapar Institute of Engineering and Technology, Patiala, and Dr. Basak, NIT Jalandhar. And some of you are joining virtually with us, and we really appreciate their enthusiasm and interest, because despite the time differences, they are with us virtually. Thank you very much. I am very sure that your contribution and insightful dedications, deliberations will truly strengthen this scientific event. We also extend our gratitude to all the delegates and participants from India and abroad. Your presence, enthusiasm, and scholarly engagement have added great value to this gathering. Our thanks also go to the print and electronic media for the entire coverage to the wider audience and the dissemination of this conference. A very special word of appreciation to the entire team of the organizing committee, and which is ably led by Dr. Himanshu Singh. And I would like to give him and the entire organizing committee and the team a big round of applause for their vision, and Dr. Himanshu, especially your visionary leadership and tireless efforts are visible. So our heartfelt thanks to all faculty, coordinators, research scholars, and student volunteers who have worked with great dedication to make this event a resounding success. As we conclude this inaugural session, I wish BioInnovate 2025 immense success in the days ahead, and hope that the discussion, deliberations, and partnerships, collaborations emerging from this platform definitely going to bring some substantial outcomes towards the sustainable development goal. With this, I wish you all the best. And students, again, my suggestion to you, please do some homework, I would say, when you come back to your school for the classes. Our faculty members, me and my HODs will ask you that what highlighted points you have noted and what is your further plan. So do not sit here casually. Please take the advantage of this scientific meet. Thank you very much.
Thank you so much, ma'am, for your heartfelt words of appreciation. With this, we conclude the inaugural session of BioInnovate 2025. We now move forward to the keynote lecture session moderated by Dr. Shashank Gurk sir, where eminent speakers and researchers from across the globe will share their expertise on diverse areas of biotechnology and sustainable innovation.
Ladies and gentlemen, we now commence the session of BioInnovate 2025. Our first lecture will be delivered by the staff scientist at National Institute of Immunology, New Delhi, India, Dr. Anil Kumar sir. He will be presenting on the topic towards investigating the anti-cancer role and gut microbiota-derived metabolites. Let's have a huge round of applause as we welcome Dr. Anil Kumar sir on stage, along with the session chair, Dr. Rashmi Rana ma'am, and session co-chair, Dr. Himanshu Singh sir from the Department of Biotechnology. Ladies and gentlemen, please give a huge round of applause for our dignitaries.
Presentation. Okay. So first of all, I would like to thank Dr. Neeta Raj, Dr. Avdesh Verma for inviting me here. It is indeed a pleasure to be here. So I am Dr. Anil, and I work at National Institute of Immunology. And today I am going to talk about microbiota, especially microbial metabolite, how these microbial metabolites are important and what role they play in human diseases and human health. Okay. Okay. So gut microbiota, if you go by definition, gut microbiota, you can say it is gut flora or gastrointestinal microbiota. It is actually complex community of microorganisms which live inside you, and these microbes not only can be found in human, but insects and in other animals, microbiota is there. So human body carries representative of all branches. So not only bacteria, but like fungi, virus, archaea, all forms of life are there inside your gut. So human is not just human, but you can say it is a holobiont. Okay, in a very real sense, humans and their microbiome forms composite organism, and it is called holobiont. So some metabolites or chemicals which are flowing in your blood are actually 10% of metabolites are coming from microbiota, from fungi or from bacteria, and they are playing a different role in human health and diseases. So this slide is having some facts. So your colon contains the highest microbial density recorded in any habitat on Earth. So you can imagine how much impact they are having on your body. So these bacteria represents between 300 to 1000 different species, but most of the species, 99% of the bacteria are coming from 30 to 40 species only. And these microbes are actually anaerobic in nature. So when they come in contact with oxygen, so they die. So 99% bacteria in your gut are anaerobic in nature. And most of the species cannot be studied outside, so they are not culturable bacteria. They are inculturable bacteria. So new field is coming, and that is culturomics. In culturomics, we can design different media, we can manipulate conditions in which these bacteria grow, and we can make these bacteria from unculturable to culturable using culturomics. So while there are a small number of core species of microbes shared by most individuals, populations of microbes can vary widely among different individuals. So we all are having different bacteria in our gut, and they are your signature. Okay, so we don't differ by genes, but we also differ by these bacteria. So these bacteria are equal to your body cells. So you can imagine how much impact they are having on our body, and their collective genes are actually 150 times more. Okay, so you can imagine 150 times more bacterial genes are having substantial impact on human health and diseases. And today, almost all kind of diseases, whether it is neuro disorder, whether it is cancer, whether it is diabetes, so all kind of diseases can be linked with the microbiome, with these bacteria and fungi which are living inside us. Okay, so my lab is studying the link between bacteria and cancer. And like not many people know about this fact, bacteria can cause cancer, okay, or bacteria can be used for the treatment of cancer. So this link between bacteria and cancer, my lab is engaged in those kind of activities. So where these microbial metabolites can be produced in the gut, so these metabolites are actually formed by bacteria, and these metabolites can have a direct or indirect effect. So once these metabolites form in our body, so they reach to different organs like heart, kidney, brain. So these organs can be affected if these metabolites are elevated in human blood or in other human body fluid. They also can interact with the intestinal cells, so they also can they can have local impact also. So cancer and microbiota. So according to WHO, cancer, like 15%, more than 50% of cancer diagnosed globally are attributed to carcinogenic infection caused by bacteria and viruses. Like Epstein-Barr virus can cause lymphoma, or Helicobacter pylori, which is quite famous example for gastric cancer. Hepatitis C virus can cause liver cancer, and HPV, which is very common nowadays, HPV infection, and HPV infection can cause cervical cancer in women. Okay, so in more than 99% cases, it is HPV which cause cervical cancer. And not only cervical cancer, HPV can also cause oral cancer or anal cancer. So there are biological carcinogens, and we are implicating these biological carcinogens with cancer causations. So researchers who are working in this area, they are wondering whether some bacteria or some bacterial metabolite can be linked with colorectal cancer. So this is the area where I am working in. Helicobacter pylori and gastric cancer. So this field is so important when we are linking bacteria with cancer causation. So in 2005, exploring this link, Professor Barry Marshall and Robin Warren. So these two scientists got Nobel Prize by discovering the fact that some bacteria like H. pylori can be implicated in the causation of cancer. So now people are exploring more bacteria which can be linked with cancer causation. So these are some bacteria: Fusobacterium nucleatum, E. coli, Enterococcus faecalis, S. bovis, S. Bacteroid fragilis, H. pylori, C. septicum, Salmonella enterica. So all these bacteria can be implicated in cancer causations. So how these bacteria or metabolites can be implicated in the causation of cancer? So if you see this slide, so TMA, which is a gut metabolite, or colibactin. So some bacteria like E. coli, PKS plus, or Fusobacterium nucleatum, they secrete metabolite like trimethylamine or trimethylamine oxide, or other metabolite which I will discuss later in my presentation. So they can interact with intestinal epithelial cell, they can damage DNA, or they can activate cancer signaling pathways, or they can make intestinal epithelial cell to secrete some pro-inflammatory cytokines. So whether single event or these events in a combined way, they can lead to colorectal cancer. Okay. So far, microbiome, which is the collective study of bacteria, microbiome has been studied in greater detail. But microbiome, which is the study of fungi, it is also coming in picture nowadays. So 2 years back, 2 or 3 big papers came in this field, and according to those papers, in almost all kind of cancer, fungi is there. Okay, so in all kind of cancer, we found bacteria and fungi in biopsy samples. So fungi is there in all kind of cancer. So it is the question: fungi is a causative organism, or fungi is there to treat the cancer? Okay, so researchers who are working in this area, they are exploring the link. They are trying to find out the answer whether fungi or bacteria, they are causative organism or they are there to treat the cancer. Or body is recruiting, like body can recruit B cell or T cell for cancer site or for the site of infection. So fact is that some bacteria are there which can cause the cancer, but some bacteria are there or metabolites are there which can be used to treat the cancer. So far in this field, when we link bacteria and cancer, so E. coli PKS plus is a bacterium which secrete colibactin, and if somebody is having E. coli PKS plus strain in his gut, then it will secrete colibactin, and colibactin will damage DNA. Okay, so we have concrete evidence colibactin can damage DNA, and this DNA damage or mutation can lead to colorectal cancer. Okay, so metabolites like colibactin. So people who are working in this area, they are trying to find out more metabolites of this nature, colibactin, like trimethylamine oxide or PCS or indoxysulfate. Okay. So far we got to know some bacteria or some metabolites are there which can cause cancer. Okay, but some bacteria or some microbial metabolites are there which can be used to treat the cancer. Okay, so 100 years back, Dr. William Coley, who was American doctor, so he used to give bacteria to bone cancer patient, and many patients got healed by those bacteria. So this field of exploring bacteria and metabolites for treating the cancer actually has been started long back, and then Dr. Anand Chakravarti did some work in this area. So in my lab, I explored 3 metabolites, bacterial metabolites, which can be used to treat colorectal cancer. So what motivated me? So in science is a big journal. So in 2021, big paper came, which highlighted the role of inosine. Inosine is a gut metabolite, and inosine modulated immune response, and it improved it augmented the efficacy of immunotherapy. So after inosine, then many metabolites have been studied for their role as anti-cancer agent. So when we talk about gut metabolites, so short chain fatty acids are most studied gut metabolites. So when we eat fibrous food or vegetables, so bacteria can convert these fibrous food into short chain fatty acids, and these short chain fatty acids like butyrate, they have anti-cancer effect. So these all are known. So in this field, like short chain fatty acid are known for their anti-cancer effect, but we have more than 200 metabolites. Today we know more than 200 metabolites which have different role in human health and diseases. So my lab is exploring whether these metabolites have anti-cancer effect or not. So far we found 3 metabolites which have anti-cancer effect. So you can wonder how these metabolites actually target cancer cells. So they can target cancer cell by apoptosis, or they can induce autophagy, or they can make different immune cells to secrete perforins or granzymes. So these are some mechanisms through which these bacteria and metabolites can target cancer cells. So these 3 metabolites, 4-ethyl phenyl sulfate, p-cresol sulfate, and indoxyl sulfate. So we studied many metabolites, but we found these 3 metabolites have anti-cancer effect. So one by one we studied the effect of these metabolites on colon cancer cell. So 4-ethyl phenyl sulfate can be formed by some bacteria in our gut, and from our gut then they can go to liver, and from liver they can go to bloodstream. So in our study we took 2 cell line. One is HCT116, which is colorectal cancer cell line, and other is CCD841, which is normal cell line. So we did the MTT assay, and we found in a very selective way, you can compare these 2 circles, and you can see ethyl phenyl sulfate is having more deleterious effect on cancer cell, which is HCT116, and it is not damaging that much CCD841, which is normal cell lines. And then we did the series of different assays, and we found like we checked colony morphology of cells, we did ATP count assay, and we found when we are increasing the concentration of EPS, then it is killing cancer cell proportionally. Then we did apoptosis assays, we checked cell cycle things, and we found EPS can kill cancer cell in a selective way. EPS can induce apoptosis, EPS can halt cell cycle. Then p-cresol sulfate, we checked this metabolite, and this metabolite, if we eat like protein rich diet, so our bacteria can make PCS from protein rich diet, and then p-cresol come to liver, and then liver can make p-cresol to p-cresol sulfate, and then again it goes to blood. And in this case we did same assays, like we did MTT assays and other assays, and we found in a very selective way it is not harming normal cells, but it is killing cancer cell in a very selective way. Cell cycle analysis we did, and we found like cell cycle arrest is there when we exposed colon cancer cell to PCS. Then we did apoptosis and Ross assay. Then third metabolite we checked, it was indoxyl sulfate. So indoxyl sulfate, if your diet is rich in tryptophan, then some bacteria are there which are having tryptophanase, and through the action of this enzyme, then tryptophan can be converted to indole. These indole derivatives go to blood and from blood to liver. Then liver is having sulfotransferase and indole derivatives can be converted to indoxyl sulfate. And you will be surprised like many cancer drugs are actually indole derivatives. So this fact is also motivated us to explore indoxyl sulfate as anti-cancer agent. Whether it is sunitinib or vincristine or vinblastine, so all these cancer drugs which are in clinical practice are actually indole derivatives. So this metabolite indoxyl sulfate was also indole derivative, and then we checked whether it is anti-cancer agent or not. So in this case, we checked anti-cancer effect of indoxyl sulfate on two different cell lines, SCT116 and ST29. So these two cell lines we took, and we found as we are increasing the concentration of indoxyl sulfate, so cancer cells are killing. We took 3 time point, 24 hours, 48 hours, 72 hours. Then we did cell titer assays. We did LDH assay, which is about membrane integrity assay. So indoxyl sulfate can damage membrane integrity of cancer cells. So as we increased concentration, so at 5 millimolar and 10 millimolar, so membrane got compromised of cancer cells. We did then apoptosis and cell cycle assays. We checked colony formation things, and we found colony formation is not there when we are increasing the concentration of indoxyl sulfate. Then we extended this in vitro study to animal study, and we checked whether this indoxyl sulfate is safe for clinical application or not. So we fed mice with indoxyl sulfate, and we checked not much difference was found like on spleen length or on intestinal length. So we also checked histology of colon, and we found indoxyl sulfate is not damaging normal colon cells. Then we checked inflammatory cytokine levels, whether indoxyl sulfate can increase some cytokines, and we didn't find much difference in the inflammation. So it is not causing inflammation, it is not harming normal colonic cell, and these three metabolites are selectively killing cancer cells only. Then in our conclusion, we found indoxyl sulfate and other metabolite, they can kill cancer cell in a very selective way. They can induce apoptosis, cell cycle arrest, they can inhibit cell viability, they can inhibit colony formations, and on the other hand, normal cells in biopsy mice, these metabolites are not harming normal colonic cells. We published this work in Journal of Medicinal Chemistry. So indoxyl sulfate and ethyl phenyl sulfate, and we are also working on PCS. So we will be publishing PCS results soon. So in last, I want to acknowledge different funding agencies: SERB, ICMR, Department of Health Research, Department of Biotechnology, and core funding from my institute. So this is all from my side. If anyone has question, so I will be happy to answer that. Thank you so much.
Check. So first of all, I would like to congratulate Dr. Anil for excellent and very informative talk.
So any question from audience side? Specifically students. So Dr. Anil, I don't have a big question, but as a query. So you mentioned you develop an in vivo model?
In vivo model?
Yes, for colorectal cancer? As you
is-
mentioned, the organs, toxicity of the metabolites and other.
Yeah, so it is called AOM DSS model. It is like a chemical model for study colorectal cancer. So when we when we give AOM and DSS, so animal can develop tumors, and those tumors can be treated with
So is there any mode of action that you put your molecule inside the colon, or other organ also affected for this? I mean, must be affected.
like
This
we
is what I thought.
okay. So when we treat tumor we induced in animals, so we give injection to IM injection, or we also give oral gavage to animals. So we don't give metabolite directly to tumor site, but either we feed animal or we can give injection of this
In
metabolite.
capsule also you can try.
Yeah, we can try. Yeah.
Yes. And sir, other question, you have mentioned in vitro your experiments, right? So in vitro you mentioned the normal cell line. Which cell line you use for this, comparison?
Okay, so it was a CCD841.
Okay,
CCD841 is normal colon cell line.
normal colon cell line. Okay. And my other question, not question, it's a curiosity. So have you compared your metabolomic profile along with the microbiome? Did you get any connection with microbiome mechanism and metabolites?
No, we didn't do that study, but we are planning to do.
Yeah, that is very great.
Yeah,
And I
we are trying to do metabolomics.
Yeah. Thank you.
Okay.
Mine is done.
Okay.
Sir, it's a great pleasure to listen to you onto the microbiota and its relationship with the colorectal cancer and all kind of other cancer. And I would like to ask just one question, that what from your side that we could do to improve our health of the gut, that what kind of things we are required to modify maybe the microbiota into the a positive sense.
Okay. So this is very good question. When we eat, we eat for ourselves, but our bacteria is also like our microbiome is also very important. So what we eat is not actually the food of bacteria. So bacteria eat different things like vegetables or garlic or onion. Okay, so they love leafy vegetables. So you know about kidney, heart, liver. So these are organs, but microbiome is also invisible organ. So when we feed our cell, also feed your bacteria, because when you and your bacteria are in harmony, then you are healthy. And if you disrupt that harmony by not feeding bacteria, then diseases like heart diseases and other diseases, cancer. So like butyrate. Butyrate is a very common and most studied metabolite which come from bacteria. And if butyrate is less in concentration, then it can cause colorectal cancer. When it is in physiological range, when it is in sufficient amount in your serum, in your blood, then it protect against cancer. Okay. So leafy vegetables, answering to your question, leafy vegetables or garlic, onion, triphala, these kind of prebiotics are very important to keep the gut and gut microbiota healthy.
Okay, thank you, sir. And yeah, we would like to.
Thank you so much, sir. And thank you so much, session chair, session co-chair, for your enriching and informative expertise that had indeed broadened our scientific perspective. जोरदार तालियां हो जाएं सर के लिए.
Ladies and gentlemen, we are deeply honored to acknowledge the presence of respected Pro Vice Chancellor, lovely Professional University, Dr. Laviraj Gupta, sir. I expect a huge round of applause as we welcome sir.
Now, to mark the formal commencement of BioInnovate 2025, we proceed to the abstract book launch ceremony, a symbol of collective knowledge, innovation, and research excellence that this conference represents. To grace this significant moment, we would like to invite on stage the respected Pro-Vice Chancellor, Lovely Profession University, Dr. Laviraj Gupta sir. We also invite our distinguished dignitaries, Dr. Zamir Ahmed sir, Dr. Ajay Soni, Dr. Anil Kumar, Dr. Shama Hirevat ma'am, Dr. Girish Gaur, and Dr. Rashmi Rana to kindly join us on the stage for this special occasion. Along with them, we request the head of School of Bioengineering and Biosciences, Dr. Neeta Raj Sharma ma'am, and the esteemed head of department, Dr. Himanshu sir, to please accompany them for the launch of abstract book of BioInnovate 2025. जोरदार तालियां हो जाएं उनके लिए. Ladies and gentlemen, I would like to tell you all that this book embodies the hard work and scientific spirit of countless researchers and scholars who have contributed to the vision of sustainable innovation. So please put your hands together for the launch of abstract book of BioInnovate 2025. Thank you so much to all our dignitaries for unveiling the abstract book of BioInnovate 2025. This marks a truly symbolic beginning where research meets recognition and innovation takes its first steps toward impact.
We would now like to request all our esteemed dignitaries to kindly remain on stage as we proceed for the felicitation ceremony. And to honor our distinguished guest, we humbly request respected Pro-Vice Chancellor, Lovely Professional University, Dr. Laviraj Gupta sir, to kindly felicitate our dignitaries. Let's begin the ceremony of appreciation with a thunderous round of applause. First, we facilitate Dr. Zabir Ahmed sir, Director CSIR IIM Jammu, for his exemplary leadership and contribution to scientific innovation. Ladies and gentlemen, please give a huge round of applause as our esteemed guests are being felicitated. Next, we honor Dr. Ajay Soni, sir, President of Association of Clinical Biochemists and Microbiologists.
Dear audience, आपकी तालियां नहीं रुकनी चाहिए, जोरदार तालियां.
Next, we honor Dr. Anil Kumar sir, Staff Scientist, National Institute of Immunology, New Delhi, for its outstanding research and commitment to scientific advancement. Next, we honor Dr. Shama Hiremat ma'am, co-founder and CEO, Lab Trident Diagnostics and Healthcare Private Limited, Bangalore. She is a visionary entrepreneur transforming diagnostics and healthcare.
जोरदार तालियां हो जाए मैम के लिए.
Now we would like to honor Dr. Girish Gaur sir, Associate Director, Rivera Labs, Mumbai, whose dynamic research in biotechnology continues to inspire innovation. And next we felicitate Dr. Rashmi Rana ma'am, Associate Professor, Sri Gangaram Hospital, New Delhi, for her significant contributions to applied research and education.
I expect a thunderous round of applause, ladies and gentlemen. जोरदार तालियां हो जाए मैम के लिए.
Thank you so much, sir, for felicitating our esteemed dignitaries. And now, to make this moment everlasting, we would like to request all the dignitaries to kindly come together at the center of the stage for a group photograph. Ladies and gentlemen, let's fill the auditorium with applause once again as we capture this moment of brilliance, collaboration, and inspiration. We request all the HODs to come forward. Dr. Himanshu Singh sir, Dr. Joydeep Datta sir, Dr. Ashish Vyas sir, Dr. Umesh Gautam sir, and Dr. Tajaswi Pandey ma'am. Ladies and gentlemen, please give it up for our HODs.
Thank you so much to all our dignitaries for joining us for this wonderful group photograph and for gracing the stage with your presence. I would now like to request all our dignitaries to kindly take their respective seats, and with deep gratitude, we extend our heartfelt thanks to our respected Pro-Vice Chancellor, Dr. Laviraj Gupta sir, for gracing this occasion with his presence. एक बार जोरदार तालियां हो जाएं. Your guidance and encouragement, sir, will continue to inspire us all to move ahead with renewed enthusiasm.
Ladies and gentlemen, please give it up for our dignitaries and esteemed guests. And now we would like to continue. And now we would like to proceed to the next keynote lecture to be delivered by the Associate Director of Rivera Labs, Mumbai, Dr. Girish Gaur sir, who will be speaking on the topic Harnessing the Power of Molecular Biology and Genetic Engineering for a Better Tomorrow. Ladies and gentlemen, please give a huge round of applause as we invite Dr. Girish Gaur sir, along with the session chair, Dr. Anil Kumar sir, and session co-chair, Dr. Umesh Gautam sir, on stage.
जोरदार तालियां हो जाएं.
I think it was a tea break. Sorry, I've encroached on that. So good morning, everyone. And this is excuse me. This is vector, this is level. I think it is open. Here it is open. Yeah, now it's open. It's open.
I would like to invite the session chair, Dr. Anil Kumar sir, and session co-chair, Dr. Umesh Gautam sir, on stage. I would like to invite session chair Dr. Gulshan sir on stage. I expect a thunderous round of applause as we welcome sir on stage. Now we would like to proceed to the keynote lecture by Dr. Girish Gaur, who is speaking on the topic Harnessing the Power of Molecular Biology and Genetic Engineering for a Better Tomorrow. Please give it up for sir.
Thank you so much. So it was to be started in good morning, but now it's good afternoon. So good afternoon, everyone. So welcome to the world of molecular, should I say healthcare, should I say diagnostics, whatever you can say. Molecular medicine, you can also say. So we all know that a small edit in a gene somewhere can actually change the whole life of a person. Okay. And it can change towards cure or it can change towards a disease. So a gene editor is very important and we'll be talking about that only. So I'll leave you to that, and I'll take this moment to first of all thank all the organizers of this conference from the lovely professional university, as well as I'll also thank the knowledge partners ACBM for giving me this platform to deliberate on the topic harnessing the power of molecular biology and genetic engineering for a better tomorrow. So we'll go through this presentation. During the course of this presentation, I'll take you through different subjects as far as the foundation of this whole harnessing the power of molecular biology and genetic engineering is concerned. Plus, we'll talk about the applications, where we are standing as far as India is concerned. Plus, what is the role of education as well as awareness in this whole domain. And we'll conclude with some sustainable development goals. Okay. So I think we have talked about sustainable developmental goals since morning. So can anybody tell me what is the healthcare sustainable goal which has been given by WHO? SDG 3. What does it say? That all of us should have a healthy life, right? So we'll start with molecular biology and genetic engineering. What is molecular biology and genetic engineering? Molecular biology and genetic engineering actually represents the foundation of modern biomedical science. What they have done is they have revolutionized the way we do diagnosis, the way we treat the patients, even prevent the disease. As we look towards a healthier tomorrow, these disciplines actually enable, we say, precision, personalization, as well as prevention, the 3 Ps that we are talking about in the next or the 21st century. Let's begin it briefly by exploring how these fields come together to reshape the whole human healthcare. So, molecular biology deciphers what? It deciphers the level at which the DNA, RNA, and the proteins work. While genetic engineering, what does it do? It actually engineers or applies that understanding to modify the genetic patterns that we observe in the humans. It actually bridges the gap between the bench and the bedside. The impact actually spans from infectious disease diagnostics to all. You have genetic diseases, you have oncology everywhere. And to appreciate that potential, we must first understand how the molecular revolution began. So it started with the discovery of DNA, double helix. It all started in 1953 when Watson and Crick actually discovered DNA. And there was a discovery of, when we say central dogma of life, it was also a kind of a mechanism that was discovered. What is the central dogma of life? It is actually when we say that DNA gives rise to RNA, it is transcription process, and through translation process, RNA gives rise to protein. And protein is the fundamental life-giving molecule. your hair color, your eye color, everything is decided by the proteins that we develop during our lifetime. So over the decades, this understanding led to technologies. What are the technologies that we are seeing today? NGS, PCRs, Sanger sequencings. So these are the technologies that we are dealing with today. So these technologies actually helped us to read and interpret the genetic code of life itself. But last but not the least, clinical implications. So actually everything what we do in lab, as Dr. Nil mentioned, he took me to my life of research where we were actually exploring many molecules for cancerous diseases. But it is not just the lab that has to do. It all has to be translated to clinical applications. Otherwise, there is nothing. What we are doing is nothing actually. So in clinical practice, this means that the disease can now be diagnosed by their molecular signature. We know there are some patterns which are related to diseases and some patterns which actually make the life healthy. So molecular fingerprints and just not their symptoms. So building on this foundation, genetic engineering emerged as the major next leap. So what is genetic engineering? Genetic engineering allows direct manipulation of genes. So what are genes basically? How many genes do we have right now? Any guess? A human body, how many genes does it have? Approximate number. 32,000. So genetic engineering allows direct manipulation of genes by inserting, deleting or modifying the DNA. Modification means there is some change in the DNA structure to achieve specific outcomes. So from recombinant insulin to CRISPR, it is all dependent on genetic engineering. We are reshaping the therapeutics by doing genetic engineering. So what is happening on the clinical side? Clinically, it empowers precision interventions because we are doing something very specific to the specific organism based on the genetic structure of the organism. So it is allowing us to do some silencing of disease-causing genes or maybe introduce some protective mutations. So what needs to be done? Let's look at the key milestones, how we achieved this. So during 1970s, what happened? In 1970s, there was a discovery on recombinant DNA technology. So during that time only, we had synthetic or artificial insulin, which was produced. Okay. PCR was innovated in 1980s, and this actually helped us in molecular diagnosis. How do we do molecular diagnostics using PCRs? We amplify the specific DNA or RNA of the organism and understand whether this is diseased or non-diseased. The Human Genome Project, how many of you are aware of Human Genome Project? Okay, so it all started in 1990s with a cost of 3.2 billion USDs, and it took more than 10 years to be completed. It actually completed in 2003. So this actually gave rise to a lot of foundation for precision medicine because we came to know genetic structures of a lot of pathogenic mutations. And in recent years, there is a big talk about CRISPR. I think all of you have heard this word CRISPR. So it empowers us to selectively target the genes and edit them in vivo. So with this history in mind, we can now understand how these advances translated into molecular diagnostics. What is molecular diagnostics basically? Molecular diagnostics refers to the techniques that analyze biological markers. It is mutations, it could be specific gene structures, biological markers in the genome and the proteome, providing critical information for disease detection and management. So it happens in infectious diseases, it happens in non-infectious diseases. Non-infectious means molecular oncology as well as genetic diseases. In oncology, the gene panels detect driver mutations that guide therapy choices also. Like in some cases, if it is a EGFR, we target the tumor based on the selective drugs. And we also ensure that treatments are evidence-based. We don't just do empirical treatments. To truly understand and harness these diagnostics, we must understand the genetic basis of the disease also. So how was this done? This was done based on what we did in the Human Genome Project. So every disease, whether inherited or acquired, has a molecular structure related to it. These molecular structures may have mutations or single nucleotide polymorphisms. And these patterns need to be understood so that we can tell beforehand that this person is predisposed to a disease or not during the course of life. So understanding these changes actually helps clinicians, the doctors, to treat symptoms, not just symptoms, but the person properly. Example given here is BRCA mutation in cancer. BRCA mutations are well-known genetic mutations. And we also know that this big example happened with Angelina Jolie, where she actually underwent a BRCA testing for understanding what is the predisposition towards having a breast cancer to her. And based on that, she got some surgeries done. So this understanding actually forms the basis of personalized medicine, which tailors care to each patient needs according to the genetic profile of the patient. Similarly, what happens, I think most of you people are aware of pharmacogenomics also. Am I right? Pharmacogenomics, how many of you know about it? No hands raised. Pharmacogenomics basically deals with tailoring the drugs as per the genetic structure. A person may not get well with aspirin or say drug X, drug Y or drug Z. So based on the genetic profiling, we need to give the person very specific drug. So pharmacogenomics basically ensures that the drug therapy aligns as per the person's genetic structure. So variants in genes like CYP450 genes, and there are other genes also, they help us understand how the individuals will metabolize that specific drug or that specific molecule. Based on that, the patient will either be treated properly or we have to taper up the drug or taper down the drug. Similarly, we also get to know in case of oncology, we have heard about specific EGFR or something like that. There is a marker called HER2 also. So in cases of patients who have HER2 positive marker during their oncology period, the patients are given specific drug called Herceptin. So the chances or the probability of getting cured based on this drug interaction is much more if it is not given. The integration of genetics into the prescribing decision defines the future for personal health care. So beyond optimizing the treatment, we can now intervene on what? We can intervene on genetic level. We know the genetic structure of the person and we can intervene based on that through what we call gene therapy. So gene therapy is nothing but actually changing the structure of the existing gene in a human. So gene therapy is a new frontier that we are working on. It actually replaces or repairs the defective genes, aiming to correct the disease at the basic level of the disease. We use viral vectors, we use CRISPRs to do that. The examples that we can think of is SMA, I think SMA is mentioned here, plus retinal disorders. For clinician, what does this mean? So what I'm doing is I'm giving you a perspective of lab also and a clinical perspective also. So for clinicians, what does this mean? This means that we can move from system control to true disease modification. We don't want to control the symptoms only. We want to just eradicate the disease, and if we are able to do that, it would be very good at the genetic level. And now to understand how this is possible, we'll look closer at one of the most revolutionary tools that has come up recently, which is CRISPR-Cas. So CRISPR-Cas is a molecular tool. It acts like a molecular scissor. We can cut from somewhere, we can add something, and that's done. So it acts like a molecular scissor. It cuts and edits the DNA at specific location. It's faster, very cheap, and it's more accurate than the tools that we were using previously. We were cutting based on our endonucleases, but not that. It's very specific tool. Clinically, CRISPR is being explored for multiple diseases like sickle cell anemia, beta thalassemia, and even cancer immunology. I think you have heard the words CAR T cells. So our next slide is on CAR T cells only. So this is one of the most impactful uses of CRISPR technology. So first of all, I think let me move to oncology. So cancer, we have heard, I think, a lot of times. This is primarily a genetic disease. We normally say it is a two-hit theory when we say a cancer is acquired by a person. Only 10% of the cancers are inherited, which can pass on from one generation to another. Most of the cancers are caused almost 90%, are caused by some or other kind of mutagens. So cancer is a genetic disease at its core. So there is a need to do a molecular profiling to understand what kind of a cancer we are dealing with. It is not just one cancer. Lung cancer can be just categorized as lung cancer, but if you go deeper, it is a non-small cell lung cancer or a large cell lung cancer. Okay. So we need to understand key driver mutations also like EGFR, KRAS, BRCA, P53 that drive the tumor behavior in these oncology cases. Targeted therapies is a new term. It is costly right now, but actually this is going to be the future when we can target the cancer based on the genetic structure of the person. Companion diagnostics is again a new term which is being used properly. So I'll just take the example of a companion diagnostics which is being used currently and is FDA approved. It is called Blueprint. Blueprint actually diagnoses a patient's tumor, or should I say breast cancer tumor, into 4 different kinds of genetic profile: basal, luminal 1, luminal 2, and HER2 type. And based on this, the patient can be treated with specific treatments. For instance, even tyrosine kinase in EGFR is being used in mutated lung cancer, so to direct the EGFR directed therapy. Same is with some triple negative breast cancers where we use olaparib, which is a PARP2 inhibitor. But beyond cancer cells, genetic engineering is also revolutionizing the immune system, which I was talking about in the last slide. So CAR T cells, which I mentioned in the previous slide. So these are the cells which are basically our immune cells. Our immune cell from the self is manipulated through CRISPR technologies to target the cells which have oncogenic antigens. So it is very precise, very safe if we are able to use it properly. This is a new therapy which is coming up. It is costly right now. It is available in some hospitals across India. So molecular methods are equally vital in combating infectious diseases also. So let's explore that in the next thing. Molecular tools detect pathogens. As Dr. Anil mentioned, there are some bacteria, viruses, you know, that they require special culture techniques. Bacteria, like atypical bacteria, they are not able to grow properly in the regular cultures. So what do you need? You need a specific tool to understand whether this patient has that kind of infection or not. So molecular techniques, when used in infectious diseases, they're able to diagnose a lot of pathogens which we are not able to diagnose in routine things with unmatched speed and accuracy. Now we have seen that happening in COVID also. So COVID was a pandemic that happened, and I think only real-time PCR based reverse transcriptase PCR was the golden, or should I say the gold standard during this pandemic to diagnose the disease. Similarly, there are a lot of other technologies which are available right now for diagnosing TB, HBV, SCV, and when we talk about sustainable goals, I think India is moving towards that when we are manufacturing our own kits. The other thing that we are looking closely is vaccinology too. During COVID, we have seen that the vaccines were developed very fast, and people were like, who develops vaccines so fast? It's nothing but it is the use of technology that we already had. We already had mRNA-based technologies. We already had editing tools. And based on that, mRNA vaccines were developed. Okay. So otherwise, to develop a vaccine, you need at least 3 to 4 years minimum to develop it. So molecular approaches also help identify biomarkers and visualize diseases processes at their earlier stage. Can we use these biomarkers in imaging techniques? Yes, we can do that. So molecular imaging actually enables visualization of biological processes at cellular level, that is RNA, DNA or protein level, to detect diseases before even symptoms appear. We can do that right now. With these innovations, I think the next step is to explore how molecular biology aids in tissue regeneration. I'm giving you all the kinds of applications that can be done using molecular as a technique to jog your minds. Because I think in morning, Professor Laviraj mentioned that we have to be innovators, we have to be entrepreneurs. So I think these all kinds of applications will help you understand what we can do with just molecular technology. So when I was talking about regenerative medicine, stem cell biology represents the interface between genetics as well as tissue regeneration. Can we do tissue regeneration? Anybody, can we do tissue regeneration? Have you seen something in news recently? So these technologies actually are offering potential cures for neurodegenerative diseases, cardiac diseases, as well as metabolic disorders. Genetic engineering actually enhances their therapeutic value by ensuring safety, reducing immune rejections, as well as guided targeted differentiation. So as we advance, I think we also need to understand synthetic biology. So these are advanced technologies which are coming up right now. What is synthetic biology? Doing something in petri dishes, synthesizing something in petri dishes. Okay, so synthetic biology applies actual engineering processes to biology, where we design some kind of organism, like we did in insulin. We designed a bacteria which could produce artificial insulin for patients of diabetes. So in healthcare, it has led to bacteria that produce insulin, synthetic vaccines, and biosensors that help in detection of markers. So this field merges biology, computation, and chemistry to create intelligent therapies, such as engineered microbes that target tumors. And on the clinical side, what we see, the clinician may get a cost-effective biotherapeutics and real-time disease monitoring due to biosensors. So to make these discoveries clinically meaningful, we also need robust molecular testing platforms, such as advanced diagnostic platforms. We have been using normal PCRs, we have been using real-time PCRs, but what about others? Can we do something very near to the patient? So there are technologies which we call point-of-care technologies, which very near to the patient can give answers, that too based on the molecular technologies, molecular techniques. So next generation sequencing and microarrays are okay, but now the thing that is turning up is lab-on-chip devices that have drastically changed the way we were doing diagnostics in, should I say, our local labs. And why have they changed? Do you want accuracy for the testing that we do? Do you want reliability of the testing that we do at the site of the patient or not? So these are matchable to the kind of technologies that we are using in labs as far as accuracy and reproducibility is concerned. So portable PCRs, I don't know whether you've seen some or not, but portable PCRs and point-of-care testings, they are bringing precision diagnostics to clinics. Now the patient doesn't need to give blood, wait for 2 hours or say 48 hours or 72 hours for the test report to come back. He can give the blood there itself in the clinic and they have the answers there itself. So with these massive data outputs, there is a big gap that is needed to be filled. What is that? How we manage the data. So this is a new thing which is coming up. It is future of diagnostics, how we analyze the data. So big data and bioinformatics is the future of diagnostics. The amount of data that we are looking at, it is humongous. And the bioinformatic tools that need to be utilized to make some sense out of it are needed. So AI as well as machine learning, I think we have heard these words since morning. So AI and machine learning are actually helping us utilize this data and understand what are the kind of patterns that can be observed in patients as well as non-patient populations. So based on that, this genomic data is paving the way for predictive as well as preventive. Can we prevent some disease? Yes, based on these we can prevent some disease. So while technology is showing us advances, we must also address, so there are a lot of questions that are raised. In case of HIV, we know we need to counsel the patient first. So, but what about the genetic diseases? Do we need to do counseling before doing just some genetic testing? Yes, but let's, before going to that, let's do one thing. Can we do this exercise here? Let's compare the human speed versus the speed of computer. We were talking in morning, no? Dr. Lavi Raj actually gave you an example of that. How much time will a computer take to compare a 60 base pair sequence to available sequences in NC databases? Can anybody answer that? Microseconds, or maybe seconds to some minutes, if it is a bigger thing. What happens here? How much time? So I'll give you the answer. You said few seconds to, maybe microseconds to a few couple of minutes. But here it is, 100 years for a person without eating, drinking, sleeping, nothing doing, just reading 60 base pair per minute. So that is what computers are doing for us. And that is where we are right now. So coming back to the ethics, legal and social implications which genetic information is giving or bringing to us is, it is actually a very important question. It is very tricky question. Nobody wants to discuss it in public, but yes, molecular technologies are actually raising profound ethical questions. Patient privacy, everybody is sharing the data. Is there any privacy there? But yes, the patient needs that privacy. His data should not be shared with anybody. Genetic discrimination based on morality, are we not doing that? We are doing that. We have been doing that. Now we have another tool to do that. So I think what needs to be done is we need to prepare some legal frameworks to protect genetic data while doing these all diagnostic tests as well as fostering innovations. So medical professionals must engage in ethical deliberation because we are the only people who actually transfer the data amongst ourselves. So these ethical questions or ethical discussions also shape how governments and regulatory agencies oversee. Because we are not doing it, so somebody from government or regulatory agencies has to come in picture. So agencies like FDA, EMA, CDSCO, they are actually playing a vital role. They are coming up with very specific guidelines where we are doing genetic testing. Alongside the regulations, we also need to look at the economic, should I say, impact of these technologies that we are looking at. Because ultimately, the impact that we are facing economically only, it's actually forcing us to do something unethical. So what is happening? So while molecular and genetic technologies, they are very costly, but if you see the overall results that are obtained, they prove more cost effectively even in the long run. So if you take a patient of sepsis, for example, a patient is administered in the hospital, he actually undergoes some kind of empirical therapy for say 4 or 5 days in ICU. A bill is raised humongously, say 4 or 5 lakhs rupees, and after 4 or 5 days, you go in for a testing, which gives you a result within say 12 hours. But after 12 hours, when you get the result, you start targeted therapy. And after targeted therapy, within 2 days, the patient is ready to go out. Can you save some money or not? When we administer the genetic testing in the first go itself, so that is what we are looking at. So rather than looking at it as a product which is very costly, we should look at what is the long-term outcome of the assay. The economic let me continue. This economic reality also affects global health initiatives, and we need to share genomic resources across the nations. So what is a global health perspective? Because we are dealing with genomic medicine, we are dealing across the globe. So genomic medicine must be inclusive. It should not be exclusive. Because if it is exclusive, we are actually excluding some kind of mutations based on the races. The mutations that are present in, say, Caucasian population may not be present in Indian population. So the whole thing should be very inclusive so that we understand in totality what to be looked at. If we just look at the data from database of Western country data, we won't be able to reach some specific conclusions. So it should be very inclusive. And there are projects run by WHO also, which is a genomic equity program, plus human genome project which was run. I don't know how many of you are aware how the genomic, the whole genome project was done. They had taken DNA from multiple sources and they pooled the DNA. So nobody knew whose DNA was being tested. So they got to know what all kinds of mutations they could have observed. Okay, so that was an inclusive kind of a thing that happened there. So there are other projects also like Africa, H3 Africa project and India Global Genome Initiative, which actually contribute our mutations, our SNPs to the global database. So that we come to know that this is the difference that we have from the Western population. So speaking of India, let us explore what India is actually doing. So India, anyways, if you see post-COVID also, we have got a lot of manufacturing hubs in molecular diagnostics. Plus, the kind of impetus government is giving, a lot of startups have come up on the healthcare front also. So India is a rapidly emerging hub for molecular diagnostics and biotechnology. BIRAC, our own facility is BIRAC facility. So we have a lot of translational programs, the Genome India project, which I was talking about recently also, and there are a lot of startups which are working on healthcare initiatives. Molecular tests like tuberculosis, which used to cost us somewhere around 2,000 rupees per test to run, has now dropped down to below 500 rupees. So that is the kind of things we are actually bringing to the table when we are actually innovating inside the country itself. India's combination of clinical diversity, because ultimately we are not just a single genomic pool, we are multiple genomic pools across India. If you go across to northeast, west, north or south, we have different genetic pools, and the kind of genetic diversity we bring in and the kind of genetic pools the database we make, it can give us a lot of information about what we are actually looking at on the genetic front. So to sustain this momentum, we need to do some education drives also. I think on the education front, we need to do some skill development also. A stronger molecular foundation is now indispensable in medical as well as laboratory front. So the new people who are coming in these fronts, they need to understand what we are dealing with. So education is one of the most important thing that needs to be taken care of. Plus, I think right now what we are doing in lab and what are the technologies that are coming up in labs on molecular front, most of the clinicians are not aware. They are not aware how to read the reports, genetic reports. So we need to do some kind of awareness activities, some kind of teaching on that side also. Sorry, I left it there. Then we need to do public awareness also, because without public support, nothing can be taken to the roots. Not all will be involved in that kind of technology. So what are the future prospects? Let's look at the future prospects. The future points towards predictive genomics, which we were talking about, protective. We also are talking about multi-omics. I think some people here are working on multi-omics. Even synthetic organ manufacturing. So there are now technologies where we can develop, say, ear, heart, kidney in the petri dishes, in the labs. So you don't need to look for donors now. We can manufacture everything in the labs and then we can transfer it. But yes, there are some difficulties right now. We need to take care of those also. So just wanted to highlight some cases like CRISPR technologies for genetic disorders. We have heard about it. Then there is a Luxturna technology, which is actually a gene therapy for inherited eye diseases. And then there is a success of Genome India project, where we have actually made a database of people across or genomes across India. So what are the challenges ahead? So despite rapid progress, we all have challenges. We have challenges in predictive diagnostics. We have challenges in preventive diagnostics. So we have off-target targets of CRISPR technologies, which we need to look at. So there are a lot of technologies with advancement we need to look at. So these are the things which we need to look at. So I'll just conclude it by reflecting on the promises or working towards the sustainable developmental goals. So molecular biology and genetic engineering form a basic cornerstone of the new medical era. So we cannot just keep molecular diagnostics apart and do clinical sciences. We are defining precision properly, we are defining preventions properly, we are defining personalizations properly. Now there is coming of personalized treatment. It cannot be one treatment suits all. Very specific treatment for very specific person. By bridging these basic science gaps and I think the lab gaps, we'll be able to help clinical care domain by transforming medicines from reactive to proactive. Right now what we are, somebody gets fear, we give some kind of a dose. We want to be proactive. And I think the future is better, healthier, and it's very good. So thank you all. As we move forward, I think let's look at this case also. Basically this is, I don't know whether you heard about it or not, is de-extinction possible? So this is what these people have done. These are 24 people. It took 18 months and one miracle to happen. A species which was extinct almost 10,000 years back, they revived the species and they got Remus, Romulus and Khaleesi, and they were able to revive it, and they are now on target to revive many other species. I think my last line would be, I want to say healthcare is changing and we are changing it for better. Thank you. Any questions?
Thank you, Dr. Girish Gaur. Excellent presentation, eye-opening. You have started from origin of DNA, starting of DNA to de-extinction, and your beautiful presentation. And you have opened new eras for our youngsters as well as people of coming in this field. And this is your Rivera labs through which you are working and harnessing this molecular biology and genetic engineering. Marvelous. And any queries from students, youngsters? Yes.
Can we have the mic? You can come here, no? You can come here.
Thank you, sir.
He was with me yesterday also.
Though many questions will be coming.
I think we can answer one or two and then...
So good afternoon, sir. So I am Amritakshi Sen. I am from BTech Biotechnology, first year. So actually, I had quite a lot of questions written on my notepad, but after each slide, everything started getting clear.
We have all the day.
No, sir. The main question that I really had was about the colossal biosciences only. It was about gene editing, and you said it.
You said it right. It is colossal biosciences.
Yes, sir. And after hearing all that you talked about and everything, I just really wanted to know, sir, if I wanted to get an internship at Rivera Labs, what are the specific skills that I would require?
This is a tricky question.
Sir, what specific skills?
I'll answer to that. I'll give you an answer. We'll work on that.
Thank you, sir. Sir, what are the specific skills that I would require? I'm not asking for it.
Rivera, no. I'm telling you, Rivera is basically a diagnostic company, which is, so this all started in 2019, where we wanted to look at the diseases in a different way. So the whole idea was to come up with molecular diagnostics only and not to go in routine diagnostics. So routine diagnostics, you have all the routine labs which are doing HBV, HCV, HIV, TB, everybody is doing that. What we wanted to look at is something which is not being looked at. I took an example of sepsis. In case of ICUs, if you see, no company was willing. Now everybody has started looking at it and they are willing to work on it. No company was willing to look at these patients. These patients are very critical patients. They need answers very fast. What we call our USP is TAT, turnaround time. So they need answers very fast, and we wanted to work in this domain. The classical picture before 2019 was that a patient of sepsis will give blood culture sample. the answer would be received, say, after 48 hours. That was a routine thing. What will the patient or the doctor do within this 48 hours? The treatment would be empirical. Nobody knows what we are dealing with. What we have done is, we have tried to give these answers within 12 hours. Our TAT is 12 hours. We provide answers from 6 hours to 12 hours, anything beyond between that. Every hour spent in ICU by a sepsis patient decreases the lifespan by 4%. If we are able to give answers within limited time period, I think we are able to save some lives. Rivara has the USP of giving these answers. That's where we are working.
Thank you so
Your
much.
question about what you need to do that. We are a diagnostic company. If you want to learn diagnostics on oncology, diagnostics on infectious diseases, because the domain we have taken is very niche. Nobody wants to work on that. The numbers are few. Nobody wants to put their hands there. It's a burning pot if you want to put their hands there. Okay? So molecular biology is the only thing that you need to learn. Post that, we can make you learn everything.
Thank you, sir.
Thank you, sir. Anyone else? Thank you. Thank you. So,
सर यहाँ पे
Yeah.
सर
yes, please.
One is here.
Ask your
okay.
question.
So sir, when you were talking about the use of AI ML in the field of diagnostics today, you told us about the power that a computer holds in calculations. But when we talk about the use of these technologies in the field of R&D, you told me that there is a large gap between in silico methods and the lab work itself. So I just want you to quantify that lab so that we have an idea that this is the amount of gap that we need to bridge as the future innovators that we are working towards.
Okay, so I'll just take a small example. Let's not go to bioinformatics, let's go to maybe a diagnostic test which is available routinely. Dr. Shama has a lab, she's doing routines regularly. But are we able to diagnose every patient? Each test has a specific limit of detection. This is a big gap. Are we able to do that? How many patients of HIV have been treated across the world till now? Can anybody tell me? Treated, I'm using the word treated. Nobody knows. Can a patient of HIV be treated? Answer.
Yes, sir.
What is the percentage?
It is possible.
A rare phenomena. It is a rare phenomena. Only 3 people have been treated. One of them is a child who was born to an infected mother, and 2 others are adults. Because what happens is, when you are treating a patient of HIV, you are trying to actually manipulate CD4, CD8 counts, primarily CD4 counts, and you are trying to pull down the viral load below a certain limit of detection. Currently, most of the assays that are available in India or world over, the limit of detection is 50 IU per ml. Can I say with surety that a patient who has come to me, who was HIV positive, and when he has come to me for detection, and I'm saying not detected, is he cleared of the virons? No. So these are the small gaps. I'm taking a small example. We can come to the bioinformatics part also. There are a lot of gaps which we need to. Right now, if you look at NIPS is non-invasive prenatal diagnosis, prenatal screening. We use the word screening, not testing. When we are doing that, we are looking at different chromosomes. Are we by surety able to comment that each chromosome detected in our NIPS analysis will be 100%, will show 100% sensitivity? No. These are the gaps which we need to fill at the lab level as well as the bioinformatics level. Because lab will do something. What about bioinformatics? NIPS is not just a lab thing. They do a testing of all the, say, 23 pairs of chromosomes or 22 XY. But there is a bioinformatics part which actually tells you whether 13, 18, 21 trisomy would be present or not. Okay, so these are the kind of gap. I can enumerate all the gaps, but these are the, you want to quantify. Quantification is typically difficult because currently maybe your sensitivity of assay is 250 IU per ml. Tomorrow you may have 225 IU, but there would be always a gap, no? Something. Okay. Any other question?
Thank you so much, sir. Now, due to time constraints, we will move on to the questions by our session chair and co-chair.
Yeah, please.
Thank you so much, God sir. Wonderful. Please.
Thank you very much, sir, for a nice session.
Thank you so much, sir, for sharing your deep insights and highlighting the role of genetic engineering in shaping a better and sustainable future. I would also like to thank our session chair and co-chair. Dear audience, please give it up for our dignitaries.
Ladies and gentlemen, moving forward, we continue with our next keynote lecture by the co-founder and CEO of Trident Diagnostics and Healthcare Private Limited, Bangalore, Professor Shama Hiremat. She will be presenting on the topic Laboratory Automation, Advancements in Sustainability. Let's welcome Professor Hiremat with a big round of applause. And I would also invite our session chair, Dr. Girish Gaur sir, and session co-chair, Dr. Ashish Vyas sir, on stage with a thunderous round of applause, ladies and gentlemen.
Slideshow. मैंने कर लिया It's there. But here it is not.
Dear audience, please maintain silence and decorum in the auditorium.
Good afternoon. Little louder. Nice to see younger generation. I usually say that you all are 4 generations difference to us. So we are much older now. You have high, for you all the access to Google is much easier. You can access very easily, but I want to start this with saying Guru Brahma, Guru Vishnu, Guru Devo Maheshwara, Guru Sakshat Parabrahma, Tasmai Shri Guruve Namaha. Guru, the teachers are very important. For you all today, I think Dr. Lavri Raj explained very well. Opportunities, when comes, you have to grab it. That is very, very important. And as Dr. Zabir mentioned, sustainable environment saving activities have to start from us, that is within and within our premises at the initial stage. That is very important. Today's topic, when was given to me by our team, in sense I call Dr. Neeta Madam also as our team, because when she approached ACBM, then our whole team worked for it, including Dr. Soni sir, the president, Shivadarsh, who is the lead man, our secretary, Dr. Girish, Dr. Alpa, and many more. I want to thank them because without everyone's support, this event was not possible. Secondly, very, very important is y'all. Without you, I couldn't be here, isn't it? So I thank everyone to be patient and listen to the talk, wherein I just want to, explain that what will be the future of diagnostic industry. So the topic is on advancement in healthcare, lab automation and sustainability. But beyond that, the question is always, right, whenever we see, I think when you go back today is a homework for you. You can see a movie called as Splice, 2009 movie. Okay, all of you go and watch that movie. 2009 and we are at 2025, right? And if anybody has watched this movie, well and good. If not, you can watch it. Okay? So with this, I think it's clear, right? So what to tell the Google students? You can teach us. So you are also our gurus, isn't it? So when we talk about healthcare, I just wanted to, stress on laboratories. How many people have gone to labs? Just raise your hand. How many of you have visited a lab? And during COVID? No one? Great, madam. So labs, a diagnostic lab, everyone has visited at least once in your lifetime, at least once, with your parents or with your grandparents, because evidence-based medicine is very, very important. You go to any physician, you go to any doctor, what they do? They ask for some investigations first, right? So evidence, without evidence, if it is a small fracture, it's an X-ray. The next level, we usually say CT. If it is MRI, during COVID, you have seen people going for an MRI also. Okay? So without evidence, the next, before giving a medications, the evidence placed is very, very essential. So when we talk about healthcare, now you may think, even I was thinking when madam asked us to give a talk for bioinformatics and biotechnology students, engineering students. So I was just thinking, how can I do this? I thought you all are much smarter than me, right? Yes? Isn't it? Your teachers are much smarter than me. But when this topic was given to me, I started thinking what to do. Until last also I was thinking and the slides were, either adding up or deleting. Means I was not able to do it in a full-fledged way. But I tried I have put a lot of efforts for this to, make you understand one thing in life. When opportunities are given, you have to grab it. The second very important thing, very, very important, slides are very few. There are more movies, more videos here, which you can watch and learn. Okay? So, what happens is, you have to, somebody asked about bridging, the gap. The bridging gap is important. If you ask me, how much is the gap? Will I be able to answer? You have to find it out. If you are, then you are an entrepreneur. You have come out with a solution. Bio-innovation, the topic for today. And one more thing today when I was discussing is about Maggie. Shiva mentioned everybody is stealing 2 minutes here, 2 minutes. 2 minutes for Maggie. 2 minutes of Maggie. How fast can you prepare? 2 minutes. How fast you can eat? 1 minute. Yes? Am I right? And preparing dal chawal, half an hour. But what are the side effects of noodles? Same way when we talk here, see the cutting edge technologies that enhance efficiency, precision and accuracy. I want to give examples because when you talk about eating, people love. When you talk about technology and other things like, people feel a little drowsy. So I thought I'd just give examples about our day-to-day activities to understand in healthcare how automation is bringing a lot of changes. Okay? So with the cutting edge of efficiency means what? When you go to the laboratory, what do you expect? Usually, you get a faster report, right? After writing an exam, you expect a faster result, right? Isn't it? Yes or no? And you expect a better result, a good result, depending on your capability. Isn't it? So if you're writing for 100 marks and you're sure you're getting 100 marks, I know nowadays students get more than 100, that is a different thing. But you're sure, so sure that you're going to get 100, and in case, I'm very sorry to say, if you get 70, 75, what do? You try to see why you have got 70, right? Isn't it? The same thing with the laboratory. The only difference is here we are, I usually say to all the staff, to all my students that we are playing with humans. So we should be very, very accurate. What is accuracy? How close you to the true value, right? Your marks is 100 and you have got 70. Is it true? No. And what is precision? Every time, reproducibility, that every time it is done, it should be the same, isn't it? So accuracy, precision and efficiency, all this when it comes together, then what happens? The results are, yes, it's a thumbs up to your results. So here, efficiency, precision and accuracy, while focusing on the subject for today, what is that? Your management mentioned, Dr. Lovely sir mentioned about is sustainable. Wow, what I told Maggie agar open kiya, packet, cover, etc., is more than what you eat Maggie. Isn't it? How much of environmental waste are you doing? Isn't it? Why I am giving this example? Because when you go to the laboratory, you will be surprised. Please visit. I have told madam, you can please visit laboratories. This is how the association is going to happen. When you visit, what I usually say, many of them call me in the, hey, what is this lady, always she finds mistakes. What the thing is, whenever I try to find the facts where you're going wrong. Is it right or wrong? How many people feel that I am wrong? How many people feel I'm right? Oh, very good. So I got thumbs up here. Okay. So this is very important, when we try to innovate anything. I will give you the last slide. It's very beautiful. I want you to answer that slide. Our main concentration should be, yes, innovation, gap, what is there? I have, developed something. But if there is so much of environmental threat, is it a right way of doing? How many people feel it is right? Because already our generation have done a lot of loss to the whole society. The plastic bags, I was very happy, to carry plastic bags. Now in my vehicle, you take a car, you take a two-wheeler, everywhere I keep a bag. I feel very disgusted because I don't want my next generations to suffer. We have already done a huge, what we can say is disturbance in the whole environment. And what do you expect? We can't. We have done it. At least we can recorrect it. As the young students, and I can call as entrepreneurs, because you see nowadays you come out with great ideas because you know that gap. Okay, as you mentioned, there is a gap. How to fill that gap? If you know this, then nothing more than that. I needn't be here at all, isn't it? So this is very, very important. So now I would like to tell on the current trends of lab, the same thing whatever I have explained. Integration of robotics. What happens? Faster, Maggie, fast, I want the things to become faster. So newer technologies are coming, but at the same time, see to that it is sustainable, not disturbing the whole environment, ecosystem. Okay, so robots are playing into the roles. Now you can see a lot of equipments coming up. I'll go to the last slide where you'll understand the whole thing, what I'm talking now. Now I'll be talking in bits and pieces. Then this is a puzzle. And then later we'll make it a single movie. Okay, fine. So then AI and machine learning, as already Dr. Girish mentioned, and there are people who are mentioning about AI. What AI does? See, after all, you all are humans. We have brains, isn't it? We are the one who have created it. So it's what you feed, you get it. I was talking to madam What we eat, we are. Isn't it? The same thing, what are you going to feed? What is your feeding is your answer. And what data is being there, the population study what we do. Okay, I'll give you the example later. On that basis, you will be able, the machine knows now that I have hundreds, thousands, lakhs of data. In genetics at least, I don't know. It's really, bioinformatics is a huge department because there's huge data on which they are sitting, and still there are changes happening because of physiological changes. DNA, what we may expect may not happen. Sometimes we say, no, it is genetic disorder. If you take a family history, okay, from him to him to him, but certain times we see that nothing is there. No family history, nothing, no background information, still you see a cancer patient, isn't it? Okay, HIV, as he mentioned, yes, you can go back and see where he might have got a needle stick, etc., etc. Okay, so even for HIV, I want to say, as sir mentioned, there is no treatment, but life can be prolonged, okay? The person who has HIV positive, whoever it is, whoever you meet, you can always explain that the lifespan has been extended, and we should thank our government because they have come out with various programs wherein they have different combinations, which is number one in all over the world. The prolonging of life is much better, which we have seen in lot of cases wherein the values, as sir mentioned, it is much lesser. So we can't tell it they are negative, but of course, the values would have come much lesser, okay? Then bioinformatic impact. So I needn't explain more on this. Then, as I already mentioned, energy consumption. When we say about equipment, when we said about the Maggie, the more the packets here and the same thing, the more we are going to technology, we have to see that it is very, very efficient. It may give a fast result, I agree. You know, every company comes and says, it is giving a best result, fast result, yes. But at what cost? They say, what, madam, what cost? What does it mean, cost? Your electricity? What about the space? Who has space? The laboratory has to spend a lot of space, right? The lesser the space the equipment take, it is efficiency, right? The faster the result without causing any disturbances to the environment. When we say environment, you believe it or not, there are, liters of liquid waste generated. For this, there is an norm as per the environment, that is pollution control. So these waste, excuse me, can you give me water? So these waste usually are treated and then been released to the sewage system. Yeah, thank you. So what happens? Can we think of something, as you said, bridging the gap, think of something much innovative there? Because biomedical waste is the biggest challenge in healthcare. Please understand, biggest, I'm giving the gap. No, the gap also I'm telling it is very, very big. No, don't ask me how big it is. Okay, I'm not a computer or not an engineer to tell you how much it is. But yes, huge, there's a huge potential. Because the liquid waste what is generated is being treated and then put it in the sewage, very clear. It has been done. But can we come out with some solutions? So there is a huge potential here, correct? Next, as I already mentioned, plastic waste. I've spoken with the first line, again the same thing, the plastic waste which is generated. If we go back to 20 years back when we started our career, so it's more than 25 years back, the PPT, everything was manual and there was so much of waste generated. And because of that, waste has been reduced. But that is not the waste has reduced where? It is only the human resource who are using the waste has been reduced. But as such, in the automation, in the system, yes, it is generated. You have cuvettes. There are tips. Okay, there are mixers, mixing tips, and many more. So these are waste, still a waste. Okay, so plastic waste is not zero, but how we can see to that this waste is minimized. Okay, so one of the solutions, see, you can see innovative solutions. I will not go into depth of this. I will share the slide. So if you have anything, you can, discuss later. So there is one point is the sources of plastic waste and environmental impact. As you, all of you know, there is impact. Everybody knows. Now somebody was telling about the grab. Everyone knows there is a problem. Yes, give the solution. You give, not necessarily that people who have a problem have to give a solution. No, not necessarily. हमें भगवान ने आंखें दी है, उसके ऊपर हम 2 एक्स्ट्रा आंख भी डालते हैं आजकल. उसके बाद भी we are not able to identify. If you have identified, this is a very good place where innovations can happen. Huge potential. You can grab it. Opportunities है, करो. Okay? And इतनी अच्छी यूनिवर्सिटी you have backed up with Madam Himanshu sir है और HODs जो बेटे हैं. See, I don't know their names, I just heard, but it's great. I told no, first Guru. Go to them, give your suggestions, come out. And nothing can get in overnight. It's only your movie tickets that you can get instant. Anything is instant, I don't prefer. Which takes longer time, the pain involved, then the result is अच्छा रहेगा. भगवान से जाके और you go to your own whatever positive, you pray and then write your exams is something different, isn't it? और just instantly cheat लेके जाओ, अगर पकड़े जाओ तो क्या होगा? No, not possible. So please be, start thinking over this. As your Dr. Sir mentioned, lovely Sir mentioned that all have to, start thinking. Passion रहना है. Main thing is the passion, which I feel it is. I'm not telling that it is lacking in anyone. Maybe you have it, but वो अंदर बैठ के you don't water it. You bring it out, ना? Isn't it? अगर अंदर grow करेंगे तो क्या होगा? अंदर से तो बाहर आना है. You have to come out. You have to. They say no, not VOMIT, not OMIT. VOMIT. What is the difference between VOMIT and OMIT? किसको पता है? Vomit. So teacher के पास जाके वैसे नहीं करना है. Whatever thoughts are there, you put it. Not omit. यानी कि अंदर है तो it will. It's like a duster, no? You just vomit it off. So we should not do that. And it is not easy. I will give you an example. Not being in healthcare also, how innovations can happen. Okay? And I'm very sorry that if I'm making you all bore, because lunch time है क्या है madam इतना बात कर रही है. So don't worry, you'll be eating more now. Because when brain starts working, your hunger levels also increases and you eat more. Okay? Benefits of lab automation as already mentioned. Have I mentioned? I mentioned about the efficiency, the productivity, accuracy and precision. Right? This is very important. No? This is very, very, very important. The power of collaboration. Whenever I talk to people, no, I am an engineer. Okay, I am a doctor. I have a bioinformatics. You divide yourself and your own friends. When you in school days, you are all were one. 1 minute का झगड़ा रहता था. Back again, all of you together. Now, why is the difference now? Why are you dividing yourselves? If you all sit together, have a same passion, have a same vision and motto, you can come out, with great ideas. I'll tell you, each and every one are entrepreneurs. Come out, bring your ideas. Very important. And ideas should be environment friendly. I'm telling you again and again, environment friendly. Data अगर हम बहुत store कर रहे हैं, what happens? I know, everybody knows what is the impact of it. Everything is getting dried. Come out of ideas wherein you are going to restore your environment. As I think Dr. Zabir said that we have so much biodiversity, we have so many plants, medicinal plants, you are going to just destroy them. How? By coming out with great ideas, great innovations without even having 1 inch of thinking towards your environment. Please start thinking about the environment. Jo bhi karo. Even somebody has put a waste. I always tell, remove it and put it. He may not do first day, second day. I think Munna Bhai MBBS kitne bhi, I think everybody have watched that movie, isn't it? So sometimes people learn by seeing others, and sometimes you have to push people. So दो लोग रहते हैं, try to understand your own people, and some people are very intelligent. Please utilize. And some people are very, very innovative. Ideas मिल गई तो आता है. In a family, you would have seen, no? If you see, पहले generation में 10-15 children used to be there and they used to run a great business. Everyone used to manage very well. That is because of different, every bird individual is different. When we don't have 5 fingers of equal number, you imagine if all 5 fingers were same, you would not be able to eat. जो हमारा जन्मसिद्ध अधिकार है, isn't it? If you eat with the spoon and fork, are you comfortable? No. वो जब use करेंगे तभी पता चलता है उसका स्वाद, isn't it? The same way, you have to come out with innovative ideas which doesn't hamper the whole environment system. This is very, very important. Okay. The same thing I have given examples here. When everything gets collaborated, successful, to become successful, you need, whom you need? Data science, you need engineers, you need doctors, okay? You need all specialized people to be there in system. It can't be that one-man show. Nahi hota hai. You want everyone to be in the system. And everyone can contribute. And now it is made in India, right? I think maybe in short period of 5 years, I hope, I will give an example here of some other country product, but I am very sorry to say, we have to develop. है हमारे पास. Again, वो इधर उधर spread है. We are like, we should not be crabs. वो story है ना, that in this one, the crabs, they pull each other and they don't, if they don't allow one person to come up, maybe as a team, he may present. What you do is, you pull each other. So he also will not be able to do, nor you will be do. So come out with the innovative ideas. You all can sit together. It is not that, bioinformatics alaj hai, wo engineer hai, we are from life science. No. All of you come together and come out with the proper solutions. Come to your teachers, talk to them, understand. Because they are not Google. Please understand this. Google wants you to be happy. Teachers don't want you to be happy. I always say. They want you to be very, very happy. Isn't it? Okay. So this is the same thing, sustainable practice in healthcare labs. I have just given. So the future directions 2024, 27 up to 2030. Automation ke bare mein, that's what the same thing with examples. So as sir said about LOT, you can see that nowadays the new technology where it is. Again chip based, lab on chips, LOC जो हम बोलते हैं. Everything can be instantly given, but at the same time how sustainable it is, how accurate it is, how precise it is, that has to be looked into, isn't it? So I will not go into depth because already have given good number of examples from the beginning. So integrated robot, see I'll tell you, healthcare industry as such has lot of statutory issues. Please understand that. बहुत सारे मूवी में देखते होंगे ना, you will be motivated that today he goes, tomorrow he goes back to the government that I want this, but it doesn't happen. So it takes time. Don't worry about that because it has, despite the tremendous advancement, robotics in healthcare faces challenges including high initial cost is number one. But agar we do it made in India products with great innovations, yes, it can be minimized. At the same, regulatory compliance is very, very important. So we have to clear all the regulatory compliance. That means, that's the reason I said from every sector you have people, then one can take off regulatory system, the other can look into the other aspects of the robots where you have mechanical, you have electronic, they can come out with great solutions for this. Then extreme staff training is required because robotics, whatever it is, it has to be trained to our system. Then integration, all this. So there are benefits of robotics and real world applications which I have put forth. But at the same time, how well we can take it forward also is very, very essential. So shaping the future, here I have mentioned 40% is the potential reduction, 60% is the faster diagnosis, and 75% is the waste which is generated. Okay. So for our vision, as I already mentioned, your efficiency should be good, precision, accuracy. With all this, you should have a faster. That is, efficiency means less human intervention and better result. Maybe AI, because many a times the reporting becomes much faster if you have AI in place. So this is the lab flow. This is more interesting to everyone. How is the lab flow? So lab flow, how we usually design is analytical. But what we work is analytical before the work starts. And analytical in the analysis, what is this? Is analysis before the analysis and after the analysis. So when you see this, you will remember from the time when a doctor has written a prescription for test, because writing a test also is evidence, right? Evidence based, when you say you need a proper test written. So identifying that is very important. Okay, so the test written from there, blood draw, what kind of a vacutainer is used, because it is particular anticoagulant, very difficult for you all to understand it, but you would have seen the laboratory whenever you have gone, when in the phlebotomy system, they will have different types of tubes. You can't use some other tube for some other testings. Please understand, it is a pre-analytical. So pre-analytical errors are highest till date, 70%. How to minimize? Yes, they are working on robots which can draw blood, but still when they are drawing, if there is a tight tourniquet added, or if the patient doesn't tell about their history, whether they are in the fasting condition, then also it's a pre-analytical variable. So pre-analytical variable is still very difficult question. Still there is lot to be done here, which is, I know some more innovations should happen. At the same time, this is the area where lot of trainings also required. Because what happens when it is trained, people see now, we had a recent instances where, why the potassium was high, what the nurse has done, she has drawn the sample, she realized it is not sufficient and mixed with another tube of the same patient, yes, but the potassium was high, which we never seen. Because she doesn't know, so training is required, right, at this stage. So we always say the skilled people should be allowed for that. So other things, in automation, we'll show a video, we can look into automation where skill is less required. Means other things can be done by automation, and skill we can still give it to our people who are in the system. Then comes, as I said, automation. Automation is much faster, and with quality controls in place, reports are much better. And then is the post-analytical release of report also, as everyone knows, it is LIS or HIS, Lab Information System or Hospital Information System. So reports also are much, there is no human intervention that I type something. Once the QC is passed, the results are out, directly it goes as a, and in India still there is a little bit work is happening wherein, the auto reporting, auto validation, still, lot of statutory requirement are there wherein they are still, not very convinced. But if that happens, then even the authorized people will get more time to have, and they'll get more quality work, because they can look into the other slides or other reports. I'll just load this video. Just listen to this. Can you hear?
Take this instance. The potassium is high. Why do we most often have problem with laboratory results?
Yes.
Take this instance. The potassium is high. Please connect me to the biochemist. I would like to talk and find out what went wrong.
Good morning, Doctor. I heard you were unhappy with one of the laboratory results. Can you provide me more details so I can help you in this?
Yesterday, I had sent my patient, Mr. Lal, name of changed, for some laboratory tests. And surprisingly, his potassium returned with a high value of 6.2 ME/Cool per liter. How can that happen? The reporting is wrong and irresponsible. How can I trust you to send more samples in the future?
I request everyone to kindly maintain the
This is
decorum.
absolutely unacceptable.
So, this is an innovation from Trident. You can just see how the innovation has happened here. The tube has to travel all the way to the laboratory. So that is minimized because the pathologist can see the slide from the center itself. So the lab will be preparing there because we are into B2B. So the sample has to travel all the way, maybe kilometers together. So instead of that, this is the faster technology is developed so that faster reports are delivered. So one kind of innovation, and it's a brainchild of a MBA person, not related to science, engineering. Okay, so this is the robot which I was talking about. See, there are things which doesn't require much of skills which a robot can do. Just see how fast it does in the lab. Kevin, just you can't even imagine. I think we can minimize on the human resource and utilize robots. Carries the samples and just see the movement. And the next video is, I thank Beckman for allotting us. There are many companies like Roche and Ortho and Beckman, who are into automation, but this technology, please look into it. It has minimized the space and is ready for accepting more space whenever required. It has given, it can do from pre-analytical, that is centrifugation of the sample, up to the reporting, everything system. Just see how much space they have reduced. So can we develop certain, things at made in India product? We have companies, but still not in all in one platform. Maybe we have automations, but pre-analytical with a build-up. See here, we can add if the workload increases, if the throughput increases, there is an option for addition. How beautifully they have designed, and this is because of the customer feedback. I'll just show the slide wherein they have taken the customer feedback and they come out with this kind of a robust facility. So imagine the whole thing getting compressed, how faster the results can be given. As Dr. Girish mentioned, our concentration should be how faster we can. And especially in the case of cancer markers, the faster, because doctors also are very busy, the faster before he looks into this, evidence is the report and the reports are released, he can start the treatment. The other two videos I had made it short, it was 3.5 minutes. So shorten it to 1 minute. If anybody requires, you please let me know. I will share the videos. Okay? See, just see here how fast, how everything together it is, including hematology, other departments also they have put forth in one single go. This topic as such, dealing in 30 minutes was a tough time, but I tried my level best. Okay, and this is what because of the feedback. I don't know whether the slide. That's okay. The feedbacks from the customers, Beckman has come out with these solutions. It's called pre-analytical, analytical, post-analytical, and the solutions for QC management. So that's how I think even we can think of something in future. This is what Sir mentioned. This. Please, I expect everyone to go through this video, which is available. How bioprinting will change the medicine. As Sir said, how fast. You can just go, Dr. Lovely, I think in the morning mentioned about the same thing, which is mentioned by Kevin. So you all please go through this video. Now the last question is for all of you. Can you please identify who is in danger? Who is in danger? A? B? How many say A? Please raise your hand. How many say B? Raise your hand. Okay. And how many say A? Now raise your hands. No one with A? One with A? Why A? Why A? No? Why B? Okay, the answer is here. What is the answer? What is the answer? The parachute is cut, so he is at high risk. A is at high risk. Whereas B, he has, though he is falling down from the hill, can you see the rope? Isn't it? It is not about the risk I am talking about. Identifying, it is not the expression. That's what I was just talking to my Dr. Shiva when in the yesterday. We are trying to see the face, means we are trying to see the problem only, but not going to the depth of it. When you analyze this, your gap, as you mentioned, you will identify, come out with great solutions, and you will be the next entrepreneurs. Okay? Thanks once again everyone, and namaskar. Any questions?
Can we share the mic?
Pre-analytical, analytical, and post-analytical, yes. Pre-analytical still remains 70% because you should understand the pre-analytical phase. We didn't have time to explain what pre-analytical. Pre-analytical starts from the prescription. No, many a times ACE is written, and our great typist or visiting counter people make it easy. Okay? Absolute is no peak out if it is asked. Right? So you got it? So first and the foremost is from the prescription it starts. Okay. Then next is when the patient go to the clinic or the laboratory. It's a very, in India we have common names, right? Yes. Maybe the best name is Kiran. Now Kiran can be male and female, isn't it? So the patient comes in, and with him there will be 4 people behind.
No, ma'am. Actually, what I wanted to ask was about the amount of waste generated.
Waste? Okay,
waste
I didn't
generated.
hear waste. I heard 3 phases. Okay.
Among
waste.
the 3 phases, which produces the maximum amount of waste?
Last post-analytical, there is no waste as such, but pre-analytical, there is a huge amount of waste generated. That is called biomedical waste. Also in the analytical phase, it's a waste generated, but there is a way every lab has come out with certain solutions, especially for environmental waste. We have to report every year, we have to give a report to Pollution Control Board. That is called how you give your, not you, your parents' income tax returns. Same way we have to give waste generation returns, filing to the statutory boards. Okay. So that waste is still huge, very, very huge. Okay. Second is at the analysis level, wherein we are talking to company to take back those waste for them and try to do some solutions for it. Because once one use. Okay, nowadays even people want people want use and throw plates. Okay, the same way here, single use, what happens? Again, waste generation. So one example I'll give you, if you go for investigations, if you have though the sample is 1 or 2 sample for one person, there are various tests involved, okay? So every test, if the cuvettes and the pipettes are being used, pipetting the sample maybe once, but there are again different departments there. Okay, we have not gone, so there the waste generation also still there, still huge. But I feel in India still we are, we are okay with that. But that is not so. We have to think of certain solutions. In our laboratories also we have tried to use reusable, but then our people are not happy about it. Because they feel that why unnecessary. So we are trying to, see that's why I told you awareness and training is very, very important for us. That plays a very important role for them to understand why we are, this much of waste what we have generated, the next generation is going to suffer. So that waste is there, but how to go to the next level, that solutions are still very less. Okay, so now some of the manufacturing companies, they are returning it back. So we have told them we will return it, you give us the fresh lot. Okay, and instead of paying, biomedical waste is not easy, they don't simply take the waste, we have to pay for them. And not less than a bigger laboratory or reference lab pays more than 1.5 lakhs per month for waste. Okay, so, but at the same time, okay, the paying is that is a cost, okay. But here, the waste generation, how to minimize it, you all can still come out with various solutions, isn't it? Biomedical waste, pre-analytical is there, at analytical level also it is there. You can work with different companies, just try to see how much of waste is generated. That kind of a project if you take, it becomes much easier, Later you can come out with various solutions.
Thank you so much.
Sorry, I didn't get the question at the base because it was...
It's okay, thank you.
Thank you so much, ma'am. Now, due to time constraint, we will be entertaining questions from our session chair and co-chair.
Very good afternoon, Dr. Sharma, Hiremat ma'am. It was indeed a very wonderful lecture. I was seeing one of the slide in which there was a mention of biological database. And if you look into as and when the any kind of research comes into play, if you talk about year 2000, when there was an upsurge of traditional knowledge. So Government of India came up with traditional knowledge database library. And when it comes to scientific documentation, INSDOC came into perspective. In last 1 decade, whatever data which has been generated because of these lab automation and the diagnosis and the gene mutations and all, what is the government saying to it? What all initiatives which government is doing by way of preserving their data, maintaining the data?
So I'll just give you an example. During COVID, yes, I don't know, the data was everything through software. It has been there with them. Believe it or not, till we have hard copies. But
it is with them, but then are they, has they opened up some research institute or where that data has been preserved?
I don't know. I think I have to ask people who are in the government setup how they do it. Because, yes, I do agree they have the data, but as such, what data we have or what Dr. Girish Lab has, or maybe Shiva who's sitting there, Neogen, he has, or maybe Sony, data is huge, it is there. But as you said, I have clearly mentioned the statutory compliance, how to use this data, because as such what data we have, we cannot use it even for research because consent has to be taken, right? But now the data is with us, doesn't mean that government has to do something, but how they are going to do it is still a question mark. But of course, if they are going to do, they have certain plans, but I don't know if anybody here has any idea about the plans of the government, they can. But for COVID, yes, they had done a great thing because it was not easy. We all were working. Actually, everyone have to thank all the healthcare workers. I know you have thanked them because everyone were on toes till night 12, 1, 2 o'clock, whole the period of almost a year. I think Girish had done a great job at that time. Though, but still, sir, I am very sorry that I can't give you the, what is the next move of the government. But of course, we can put forth, as you said, the data, whatever is there, but it should go through proper channel. I think sir can add something here. because still we are into private sector and we have so many norms. Even a patient consent or even patient's data is not been related to anyone. Yes.
So basically, I would like to add little points in this. At CSR level, we are running one health cohort at CSR level. Pan India we are doing, and this is restricted to CSR employees and pensioners only. So like you mentioned, whenever we want to use that data, so we need to get the prior consent from the patients and who were the subjects for these database. In India, like government organization, ICMR is the agency, like we have WHO. In India, ICMR is the authority which has to use such data for policy making, for further use in order to guide the government on different fronts. So here what we are doing, the proper format, proper procedure is adopted to use that data without disclosing the identity of the subjects. And as per the government now, certain parameters are there and we need to follow. As private organization, I think you must be following that also. So another point is that whatever data we are getting, so we need to see whether this will be useful for, because this sample size is very critical in this.
Yes.
In our case, we have the cohort system and we are getting samples from employees and pensioners. Still, we are observing whether this sample size will be useful for ICMR use or not. So that is another important issue in this.
One more thing which I want to mention, sir, that WHO has, from UN, they have one program for pregnancy, maternal and also the child. For that, they have one program which is FT4 TSH, and now they're doing B12 and vitamin D. Of course, that data is with them, and we at Trident, we are serving around, presently we have got few districts. There are 25 districts which we will be getting shortly. And the whole data will be there. And as per government, they want to do statistical analysis. 5 years ago, we had done this and they had got an award for it.
Excuse me, can I answer this? Sorry. So, excuse me this side. Hi. Hi. So, I'm Rashmi, I'm from Delhi, Sir Gangaram Hospital. I'm a WHO fellow also. So, what happened? So you must be heard that there is a funding agency called as ANRF recently. So Dr. Shiv Kumar visited our Gangaram to know what type of data you have, depository. So recently we conducted this AI in multi-omics data integration and analysis. So right now they have opened one call. Recently you can also see the call is AI Mahamaya, something like that. And I have one BioRec project in AI, because I will show you in some slides that how AI use in a larger cohort of the data, and that data will be used by other people also in context of the patient diagnostic. So this was their question. Another in context of WHO, so I'm working on dengue also, which I will show you how our data. So in ICMR provision, ICMR WHO some MOU. So I have some running project in dengue. So they ask a data, and they are making the depository and they submit to WHO. I don't know what context, but yes, they are doing that. It is going on. It will take some time.
Yeah, but the context is there are so many private labs.
Yeah.
But at the same time, the projects what are involved are at a very lesser scale.
See, ANRF giving the opportunity to the private labs also. I have a MOU with one of the company from Odisha, Imaginex. So they are making us LFA kit of dengue for the early detection using blood samples, so that they can distinguish between the severity and the initial febrile phase. So now ANRF is this is done by Dr. Jitendra Singh. He is our Science and Technology Minister. So and our CEO Dr. Shiv Kumar. They have opened it you can go through just once.
Yeah,
and you can also collaborate some government agencies and government institute. I am also a private institution, trust institution in Gangaram. So they have given this opportunity.
Yes, as you rightly mentioned, because I cannot disclose certain things, but whatever the statistics was given, there was a drastic improvement on the mother and the child care.
the level of this one at Karnataka, it's been given there wherein they presented it, but we had the data. What they asked us to do all the statistical analysis and give the report. So DHO, Dr. Srinivas got the award for it. So that was one. But now what sir mentioned is, we are sitting on so much of data, what are you going to do? But once we get some kind of
centralized kind of a data center which is to be made.
presently, but of course, see now the reference values what we are giving, we have to do a lot of work because we all are sitting on the, all the data, which has to be, channelized in a proper way, as you said. Because for this, we need all the laboratories to get intervened with the government. I think Girish would like to talk.
I just want to add something. Now we need to understand what kind of data we are collecting. During COVID, what we were collecting is positive, negative data, and it was a demographic data. Similarly, there is a portal called Nikshay. Indian government is collecting data for all the patients who have been declared as positive TB and rifampicin resistance. They are collecting the data and they are actually using those data to devise the policies where to give what drugs. So they have TB programs that are running across India. So they are doing that. Similarly, there are data which they collect through some mechanisms in the CMO office. They collect the data and they collect data for all the kind of influenzas and other diseases. So these are the diseases which are infectious diseases, and there is a mechanism by which the data is being collected. Like in case of HIV also, any HIV positive patient has to be declared, because by that way they are able to somehow follow up with the patient. Now comes a question of non-infectious data. When we are talking about exome sequencing, when we are talking about whole genome sequencing, when we are talking about maybe some kind of mutations, HER2, and the biggest problem across India that we are facing is that majority of this data is with private companies. I was just sharing with him that I think 10 years back, I was talking to Ram also, and we were thinking of making a database where we could actually use the data for public usage. But what happens is this data is a gold mine. No private company wants to give this data for public use. So what Government of India has done is, I had mentioned in India Genome Project also, so they are coming up with some mechanisms, and I cannot name, so I'm sorry I cannot name these databases that they have prepared, but they are surely working on that. And there are few private companies also which are making huge databases, because ultimately these databases are not easy to maintain. These databases need a lot of cooling, lot of infrastructure, and to tell you, they are being made in environmental issues also, and they are being made inside the oceans. And there are bigger private players who are coming up in this. So Government of India, no doubt, is collecting some data. They have made some databases, and how they're going to use it, I still don't know, but they are open to public. Not much data is available, but yes, there are some projects like India Genome Project, which are the data for which is being collected.
Recently, even biotechnology, technology as such, the whole team had been there to discuss on the same thing.
Yes, and they want to do one thing, that they want to actually have data from all the population sites of India, so that once we have this data, the bioinformatician can actually challenge this data with their own data and come up with some solutions. Like currently in genetics, we devise a mechanism where we say that this mutation is pathogenic or non-pathogenic or something. There are 5 different differentiations that we do. So today, maybe some of the mutations are non-pathogenic, but after we have functional studies down the line, maybe 1 year down the line or 2 years down the line, this same mutation can become pathogenic. So the kind of gap thinking that they were asking, that also is filled up slowly and steadily. So for sure, Government of India is doing a lot of things. Private players are being involved, but right now where they are storing the data, that to pinpoint that is difficult.
And it takes its own time, but as such, we are on data, huge data.
Thank you, Doctor. If nobody has another question, then I think so can we just pass on the mic there?
Hello. Good afternoon, ma'am. I'm Dr. Ashish from Biomedical Engineering Department. Ma'am, we are talking a lot about the AI and data. That's great because continuously from last few years we are working on that. But my question is related to the machines. You are running a very good lab. If we talk about the medical instruments, all are so much costly. And means within few, if I talk about this one, means within few months, within few years, we get a new model of the machines. Okay, and with very good accuracy and with very good precision, and a lot of new features has been introduced in the new models, new machine. So, if we talk about means what is the life of a machine? Means suppose I'm going to be buy a machine right now today. So how many years I can use that machine? Means if we talk about the doctors, they wants to be go for the side of the means with good imaging, good data, and good quality. Okay, means and if we are getting a new machine, so obviously that will provide a very good image as compared to the older one.
As a referral lab, we usually don't buy the equipments. We go on a reagent rental model. Okay, so we have a scheme wherein we never go for purchase because this is the main reason. Okay. If it is doctor driven, usually we have seen many doctors, they purchase the equipment. But as a biochemist or as a microbiologist and they pathologist, they may never go for a purchase of equipment because as you said, the technology changes. Okay. The efficiency, the throughput, today you may be running 100 samples, maybe down the lane after 4 years you will be running 400. It happens with us itself, okay. We started with 4 staff, now we are 300 employees, okay. And we started with the smaller versions of equipment, of course automated. Again, it was semi-rental scheme. Now we have everything on rental. We have different, all kind of equipment because all eggs, I mean, in the same, there is a saying, right? All the eggs in the same basket is a different thing. We need to have different technologies. See, if you say we have markers will go into, I'll just give an example of our own. It goes onto the Biotech platform. Then there are Perkin-Elmers. Roche has, but they don't have double marker. They have only double markers. They don't have this triple and quadruple markers. Okay. So technology is growing at a very faster rate. At this stage, if you go for a purchase, it costs huge. Yes, yes. And you will be spending, and by the time you get return, it is impossible that you will get the returns. That is very important. Though the doctor want everything, but at the same time, the cost involved is huge. Yes. So in that case, if any possibility, you have to go in for reagent model so that you can have options and you can close the agreement anytime. Okay. Even if you are not giving the good number of load also, they will take back the equipment. But that is a better model than going for purchase of equipment. This is what usually we say. Otherwise it will be dumped. We have seen certain labs, they will be dumping the equipment. It's like a museum.
Yes, yes, ma'am.
Every 3-4 years. Lifespan is a different thing. Here it's not more than lifespan. Yes. It is how the throughput of the equipment, and main is throughput and precision. As you said, precision and accuracy plays very important. The faster, the better. And second thing is the volume. Yeah. So the technology, the nanotechnology, the next generation equipments are much better. And the involvement of staff also will be much minimized because automation will be very high. Instead of having a robotic hand of 1, it will be having 4. So simultaneously it will be working. So I have shown the backman equipment, simultaneously it will be working multiple things. And now it has a platform from pre-analytical to post. If you have good workload, then you can go with the whole system. So that depends, but only upfront purchase will not serve the purpose. It's only in R&D section wherein you need the equipment to work for a longer time. Yes. But here it is not. So because here the service part is totally different. Here patients pay for it and they want a faster and a better report. So in that case, purchase is not a right way of choosing it. Maybe rental or also semi-rental would be fine.
Thank you so much, ma'am, for your wonderful presentation. I would also like to thank our session chair and co-chair. And now, ladies and gentlemen, please give a huge round of applause for our dignitaries. And before we move forward, we will now pause for lunch. We request all our guests, speakers, participants, and our registered students to join us for the lunch arrangements which are made in the basement of Block 29. Kindly return to auditorium by 2:30 p.m. for the continuation of our post-lunch session. All the dignitaries and registered students. I will again mention that registered students are required to return to auditorium sharp by 2:30 p.m. as their attendance will also be taken. Thank you everyone. Enjoy your meal. You have to mention that.
Sweetheart, when you're present.